So good morning and welcome to Plymouth University's Marine Station. Thank you everyone so much for coming. We really appreciate the effort, especially those of you who've travelled far and from across the channel. It's really great to see everybody here today. My name's Emma Sheehan. I'm Associate Professor of Marine Ecology here at the University of Plymouth. It's been my honour to lead the Fish Intel project and also to chair this conference today and the Pint of Science event this evening. Housekeeping. So if for any reason we need to evacuate this room, there are external stairs out those doors there, or please go back the way you came, and then we're to meet outside the gate of the Marine Station. Facilities, if you haven't found them, are back through the foyer and on the right. And also do make use of the balcony out here. This is an amazing place with a really nice spot on a beautiful sunny spring day. So please do make use of the outside space as well. This is a fully hybrid bilingual conference. What could go wrong? So please use the headsets when needed. You will need the headsets also for, we have an online speaker and there's another one as well, which I can't remember. Later on, we have a guest speaker as well, speaking from Canada. So we won't have speakers in the room. So this is for the online Zoom attendees. So we've got a really exciting packed lineup of presenters today. So I had better stop talking and get on with it. This morning, we have a series of talks with times for questions before the break and before the lunch. So if you have any questions, please keep them to yourself and then raise your hand at the question time. But we also have a Slido app. So if you look at your handout on your seat, there are some instructions about how to use the Slido app, and you can post questions on there as well. But I'm very happy for you to use the old-fashioned way, and we can have a nice question and answer session, as I said, before the break and before lunch. After lunch, we have two short workshops where we want to hear from you on how best to incorporate new research into ecosystem-based fisheries management. And we will also be providing lessons learned about fish tagging and tracking. And we'll be briefly presenting the project protocols and outputs so that you know what's been produced on this project and where to find it. So, we're on time. Fantastic. To help set the scene, we are very honoured to be joined this morning by Miranda Krestovnikoff, who is a biologist and wildlife expert with extensive knowledge of conservation environmental issues. She has presented on many BBC TV and radio shows. She is the Vice President of the Royal Society for the Protection of Birds, patron of the Whale and Dolphin Conservation Society, and patron of the Alderney Wildlife Trust. So thank you very much for joining us this morning. I'm going to hand over to you.
Morning, thanks. I'll just lower that microphone. I'm always shorter than everybody else in the room. Good morning, everyone. Lovely to see you on such a beautiful day. A special welcome to anybody joining us remotely and from outside the UK. Thank you to our interpreters who are going to be translating today. I hope I don't speak too fast. I have a habit when I'm passionate about things of sort of going at 100 miles an hour. So I'm going to try and slow down and give them a chance to translate everything that I'm saying. I've come rather late to the party for Fish Intel. I only found out about the project just earlier this year, but nonetheless, I've been learning fast about what you've been studying over the past 2 years, and it's such a pleasure to be here to celebrate the end of this fantastic project and all that it's achieved. So if I haven't been involved in this project from the start, why am I here? Well, this is me in my happy place underwater. I'm a diver first and foremost, a TV presenter second. I was lucky enough to work on the BBC's Oscar-winning series Coast way back, if you can remember that. Maybe the older ones in the room remember that far back, along with working on projects on The One Show and Radio 4's Costing the Earth and Living World series. And as Emma said, I've worked with numerous marine conservation organisations, Project Seagrass I'm getting involved with now, Marine Conservation Society on their marine protected areas. Great news, we've got 3 new ones this week. Back when I learnt to dive, I never thought that it was going to be part of my career. For me, it was just an interest and a hobby. I learnt to dive here in the UK, critically, and that's where my heart really lies. This is where I do most of my diving. My children dive here. And I dearly love the slightly murky, chilly waters around our coast. Don't ask me why, but there's something about them that's really special. I vividly remember my first few dives right here in Plymouth as a teenager on the shipwrecks. I see there's a picture of a couple of them, the Scilla and the James Egan Lane, just outside here. These are magnets for wildlife. They're stunning wrecks to dive around. I did my HSC professional scuba diving course right here in Plymouth, and I've literally had hundreds of incredible adventures all along the channel, including the Isles of Scilly and even a few weeks diving the coast of Normandy and Brittany. Now, for me, it's really interesting talking to non-divers, non-marine biologists about life underwater. People always ask, well, what do you see under there? It's hard to know where to start, really, because it's all so incredible. But I just wanted to share a few highlights from the channel from my 30 years of diving. It's not just fish and marine mammals, some invertebrates and the habitats that they live in as well. Many of them are in areas with special protection, which result in remarkable marine wildlife. To take the humble, common cuttlefish, many people are surprised when they realise how beautiful and how clever they are, when mostly all you see of a cuttlefish is the bone that's left on the beach. But we have cuttles who visit our southern shores every year to breed. Their mating display is just stunning. They have colour cells in their skin called chromatophores, and the males use these to create a dazzling display of mesmerising ripples to lure the females in to mate with them. They've already been seen near Weymouth this year, so I'm very much looking forward to some more encounters this summer. Fish species don't get a lot of kudos for their appearance here in the UK, but how beautiful are the markings of this tompot blenny? What a charismatic looking fish. When people say fish aren't charismatic, they are. The male tompots have head tentacles, which look a bit like antlers. The bushier they are, the more attractive they are to females, and the more intimidating they are to rivals. And they live in deep crevices on rocky shores and in and around shipwrecks. And the females, having chosen their favourite mate, will lay their eggs in these little gaps. The males fertilise them and guard them with their lives for a couple of months. And through decades of study, it's been realised that you can identify each and every fish by their facial markings, by identifying individuals, and you can build up an idea of who is mating with whom and their success rates in the breeding season. Also pretty fish. This is a corkwing wrasse, a male, and these males are brightly coloured, but they don't just rely on these bright colours to attract the females. You can actually sit and watch them going backwards and forwards with pieces of seaweed, making nests to attract females. I've actually sat there and offered little tiny pieces of seaweed to a male corkwing wrasse, and they take them and then they promptly spit them out and reject them. Clearly, I don't have the eye for what makes good nesting material for a male corkwing wrasse. But nest building is obviously very important to get right. The female, if she's impressed by what she sees, she'll lay her eggs in the chosen nest and the males will fertilise them. Seahorses. We are... Oh, is it going to go? No, we stop. Okay. We've two species of native seahorses here in the UK. Most people I talk to are blown away by that. Famously, seahorses, the males are the ones that give birth. This is a spiny seahorse, a resident in Studland Bay in Dorset, where they're tagged, monitored, and the habitat that they live in, the wonderful seagrass. It was World Seagrass Day yesterday, I think. That not only provides a safe space for juvenile fish like the endangered ungulate ray, it helps protect our coastlines from erosion, keeps our oceans clean, and also stores carbon. But this precious seagrass habitat is damaged by, by boat moorings, which drag and scour the seabed. And there's been a battle going on in Studland for many years between the locals and the boat owners and the conservation groups, eventually resolving in the installation of several environmentally friendly moorings, which are an alternate to anchors, designed to protect the precious seagrass. And this is an example of years and years of monitoring and data helping in the end to protect valuable and fragile. So you saw the grey seal. Some of my best grey seal encounters have been in the channel around the Isles of Scilly, where these wonderful, inquisitive mammals seek you out and seem to want to play hide and seek with you. There's nothing quite like being face to face with an intelligent, air-breathing, social mammal who's trying to work out who you are and why you need all this crazy kit to dive when they're just holding their breath and swimming around underwater. It's a real thrill to have this sort of interaction with a wild animal, something you just can't engineer. But when it comes to marine mammal encounters, this is probably my best one. I was diving on a shipwreck called the Volney, not very far from here, filming for the one show, talking about the marine life that starts to inhabit the shipwreck as it goes down. Here you can see there's a beautiful pink sea fan that I was talking about. And in my earpiece that we have when we're diving underwater so we can communicate with each other, I could hear somebody shouting something and I couldn't really hear what it was. And then I turned around and there was this wonderful solitary bottlenose dolphin who'd seen the divers in the water and very much wanted to come and play and interact with us. Now, I abandoned the sea fan. I knew that that was going to be there on the next dive. The dolphin probably wouldn't be there. I'm very much a look but don't touch sort of person. But when the dolphin sort of turned over and presented its belly to me like my puppy would at home, I knew that I needed to just gingerly stretch out a fin and give it a little bit of a tickle on the tummy. And it was a wonderful moment. It wanted to interact with the divers. We had tens of minutes of fantastic interaction with this incredible creature. So hopefully that gives just a hint of why I love diving in the channel and the diversity of marine life that we have right here. So back to why we're all here today. This project is all about understanding where different species live and where they travel to and what habitats they're using. And using that, we can understand about their preferred habitats and we can take measures to protect them. We need data on these places so we can build this information into conservation and management plans in the future. The telemetry used can follow different species and their prey species. And this incredible technology is allowing a real insight into the lives of these animals that we wouldn't otherwise get. They're animals that don't recognise international boundaries. So international collaboration is very important to meet conservation targets on a global scale. It's wonderful to have so many stakeholders, many of you here in the room today, involved in this project, having fishing communities working alongside scientists, sharing what they want to see in terms of protection. It's a really exciting project that I understand doesn't end here, that more money has been awarded for tagging of elasmobranchs. So this collaboration and these relationships that you've formed can continue forward into the future. Now, more than ever, we need to work really hard to safeguard these precious waters and habitats, providing safe havens for our marine wildlife. Marine ecosystems, I'm sure you know, are under a huge amount of pressure from all sides. So by working together with neighbouring countries to form a network of protection, mobile creatures will benefit from the security they deserve well into the future. I'm really looking forward to hearing more about the project and the results from the following speakers this morning. Thank you all very much for having me here.
Thank you very much, Miranda. That was really wonderful and very exciting. I'm going to go home and service my dive kit, I think. Yeah. Okay, so if you do have any questions for Miranda, so there's going to be a question session before the break. Right, now you're going to need your headsets. So to set the policy scene for Fish Intel, our next speaker is Pierre Karleskind. I can't say that surname. Katia tried to teach me yesterday, but I failed. Pierre is the chair of the Fisheries Committee of the EU Parliament. He's an oceanographer. He became serving as Minister of the European Parliament since 2019, and in 2020 he became the chairman of the Committee on Fisheries. So, Alistair, over to
Ladies and gentlemen, dear friends, I'm unable to be with you here today, so I'm sending you a message that I recorded in Strasbourg at the European Parliament here. I know you've worked on it for several years, thanks to an Interreg program, and I will come back to that in a moment. The channel separates France and England and the UK, but it also unites us through numerous activities. They take place there. Of course, fisheries, that's essential to the work you've been doing, but also shipping. Now and in the future, of course, wind turbines and wind farms. And they're all around the coastline. There are cities and activities that create discharges that can pollute it. And in the summer of 2022, I raised the alert about untreated sewage being discharged or released into the channel in the North Sea. That's on the British side, but of course we must recognize that on the French side, it's not that much better. You have looked at how in this sea the various species that provide value for fisheries find their place in their habitats. And if we want to go further than the current approach, essentially based on reaching the maximum sustainable yield, MSY, we have to look specifically at where these fish live. Now, we know that many species are born on the French side and then they grow up on the UK side. And all this needs to be studied. Very often in the European Union laws and in EU funding for fisheries, the first point which is addressed, what's given priority, is knowledge of the environment. We've said and repeated that we know the surface of Mars better than we know our own seabeds and seafloors. So we have to progress. We have to define which habitats should be protected, how fishing gear interact with habitats, and there's how to better manage fisheries and to have better results in terms of fish stocks, the good health of waters, and this on both sides of the English Channel. Now, I said the channel both separates and unites us. So for many years, when the UK was still a part of the European Union, we implemented the cross-channel Interreg program, Interreg France Channel England, excuse me, which made it possible, in fact, to develop numerous projects. I'm especially proud to see that your project is reaching its conclusion in term, because before 2014, when the last long-lasting Interreg program was established with the British, I was vice president of the Brittany Regional Council and actively participated in defining the objectives and goals. So today, seeing this project, which is reaching its end with Brexit, of course, our fishermen are still fishing in the same waters. Our interests are still the same, and they still apply. We need a sea with a good environmental status. We also need this sea because economic activities are taking place there. And our societies, both French and British, rely on that. So I hope that we'll be able to continue our work. During the early years, the first post-Brexit years, our relations became complicated at times between our countries and between Europe and the UK. But I think we will be able to touch land here, because what you've done should be pursued. I and my EU colleagues are particularly attentive to this, and I will soon meet a special envoy from the UK in charge of these subjects. We can't stop now, having come so far.
Okay, thank you. I hope everyone can figure out the headsets okay, though. Okay, our next speaker and the first speaker from the Fish Intel project is the wonderful Lucy Hawkes from the University of Exeter. And she's going to introduce you to the Fish Intel project, the ambition of the project, and how to set the theme for all the following talks this morning. Over to you, Lucy.
Must try to be wonderful now. Wow. It's going to be quite a pressure. Okay. All right. So, so my name's Lucy Hawkes. I'm from the University of Exeter. And I wanted to first of all tell you that we, Fish Intel, are actually a collaboration of 12 different organisations who together, under Professor Sheehan, under Emma's leadership, have been brought together, and we all share a really strong passion for fish, for fishing, and really importantly for sustainable fishing and sustainable profitable fishing into the future. Some of us are academic organisations, some of us are NGOs, charities, private organisations and fisheries representatives. And Interreg is a programme that's really been designed to bring people together. And some of us worked together before, some of us hadn't worked together before. And so one of the really great outputs from this project has been a real strength of collaboration that we hope will really lead to change in the future and for things to get really a lot better. Fish Intel principally aimed to do kind of three things. One was to increase the proportion of transitional sites with good ecological status. I'm going to tell you what those terms in blue mean in just a moment. And we've done this by identifying the most important seabed habitats for a range of different species. And this eventually allows us to operationalize something known as ecosystem-based management. And again, I'm going to tell you what that is in just a moment. So first of all, what are transitional sites? It's a proper term. It's basically anywhere where the land transitions to the sea, as you can see here. So we're talking about rivers, estuaries, salt marshes, and even some coastal waters. I also need to tell you about the Water Framework Directive. This is the legal instrument that protects European bodies of water. It provides for management recommendations. It sets regulatory frameworks. But importantly, it's the instrument that's defined this good ecological status that you saw on the previous slide. So what is good ecological status? Well, for a habitat to have good ecological status, it has to have only slight variation from undisturbed conditions. That's the official terminology. So basically as good as natural. And it's an ecological status. So it involves all the different parts of ecology. So not just the habitat and the water itself, but the things that live in it too. So the fish, invertebrates, plankton, and even things like water quality, because of course these things living in the water both influence the nutrients and the pollution in the waters as well as create those nutrients as well. So that's what good ecological status is. Now, I thought that probably quite a lot of waters probably would be in pretty good ecological status. Looking out at the water today looks lovely. But actually, this plot here shows you the proportion of waters that are in bad, poor, moderate, good and high ecological status. And what you're seeing is unfortunately far more red, orange and yellow than we'd like to see. And these are split into coastal waters, transitional waters, lakes and rivers. What I want you to notice first of all is that transitional waters has among the higher kind of amounts of bad ecological status, and that taken together, transitional waters actually have much more bad, poor and moderate waters and much less good and high ecological status than those other water bodies do. So that's why we're working in transitional waters particularly. So if we also have a look at where these ecological statuses are mapped, here what you're seeing as colours get darker and darker to red, we have less and less water that's in good ecological status. And you can see that the Channel Manche region actually has a lot of red. So it's a place where water quality could certainly be a lot better, where ecological status could be better. So that's why we're working there. To do this work, we need help. So we've recruited our fishy friends. We have 5 actors helping us in this project. The first is the European sea bass, which I know many of you in this room will know far better than I do. These can be quite big, but typically about 50 centimetres. We're getting them in estuaries, lagoons and coasts, so they're definitely in these transitional waters. They're also an important species for commercial farming as well. Then we have the pollock. Similar in size, perhaps a slightly lower trophic level diet, so mainly feeding on things like planktonic crustacea. These species are interesting because they're thought to be a much more sustainable alternative to cod. So you've probably had pollock and chips in a fish shop before. We then have the black bream, hopefully, in a moment, which is a little bit smaller. The black bream feeds again even lower again, so it feeds on algae and invertebrates. Fun fact about the black bream, they're all born female, and it's only the largest females when they get to about 40 centimetres and then sex change into males. Pretty exciting stuff. We then have my favourite, because this is the one I work on, the Atlantic bluefin tuna. Massive fish, very exciting, live in pelagic waters. These animals are zipping across the Manche, across the channel constantly, connecting transitional waters. And they're pretty high-level predators. They have very few predators themselves, although they have been known to be eaten by orca, which is very exciting. I can tell you all about that later. And these are a commercially valuable fish, but they're also prized in the recreational sector as well.
And then finally, we have crawfish, which are for obvious reasons included in this project because they're exciting, they're valuable, but they're also of conservation status, of an important conservation status as well, with strong limits on their. So these are our 5 species that we're using to tell us more about the ecosystems across the channel. So these, all 5 of these species are important components of coastal ecosystems. They represent a range of different trophic levels. They're also very important in making these good ecological status assessments, and they're all commercially valuable as well. So we think we've chosen a good bunch of collaborators to work with us. We've worked across 7 different pilot sites, and you're going to be hearing people later today talking about all 7 of these sites, 3 in the UK, 3 in France, and 1 in Belgium. And what have we actually been doing? What is this? How do we find out the answers to our questions? Well, we're using acoustic telemetry. And as the name suggests, it's using sound to transmit data. So acoustic telemetry comprises 2 parts, and I thought I'd tell you a bit about this quickly, because it's really fundamental to everything else you're going to hear today. First of all, we attach an acoustic tag to an animal, and there's actually one on display at the back of the room that you can have a look at a bit later. And these tags are sending out a sound message, which is heard by a receiver, and there's one of those at the back you can have a look at as well. So every time these fish swim within a few hundred meters of these receivers, the receiver will hear its unique identification number and log the date and the time that it saw it. And you can eventually tag loads of fish, and loads of them are swimming around, and loads of them are communicating with these receivers and telling you where they were. It's very much like, I was going to say like an Oyster card, but these days when you go to London, you just blip your bank card on the turnstile, but you know what I mean. Anyway, this is what acoustic telemetry is. And most of these tags are surgically implanted into fish. That's what the one at the back of the room would be used for. And this is what that looks like going in. But you can also attach acoustic tags to the outside of animals as well, which you can see here on this crawfish. And also this bluefin tuna is wearing an externally mounted acoustic tag, as well as another type of tracking tag that I'd be happy to talk about as well later in the coffee break if you want to grab me. And the receivers, when they go on the seabed, have to go into a special little jacket, which again is on the back of the room here, including 3 orange buoys, which help it to bob back up to the surface. These receivers have to be serviced. We have to check the batteries. We have to download the data. So we need to get them back again. And that's what this whole kind of contraption is that's set up around it. And we put these receivers all across the Channel region. These blue blobs here are showing you the locations of the Fish Intel network. And this network is really a globally significant piece of work that we've been able to contribute. It doesn't just pick up Fish Intel fish, it picks up any fish that have been tagged with acoustic tags. So it's really, really cool. And we're also supported by other people's receivers as well, which you can see in black here. So other projects in the world have receivers on the seabed. And if our fish swim past those ones, we'll hear from those too. Altogether, we've been really busy. We've tagged nearly 1,000 fish, and we put about 190 receivers on the seabed. So we've been really, really busy. I can't tell you how much work goes into each one of those tags and each one of those receivers going onto the seabed. You'll see some of these numbers might change slightly between presentations, and that's because when we service the receivers, when we ask them to come back up to the surface to download them, and as many of you who might be fishermen would know, stuff don't always stay on the seabed. So sometimes we lose some, sometimes we have to introduce new ones. So this number can be quite variable between some of the presentations you'll see today, but we've been busy as the point of this slide. And it's working too. We're receiving detections from the fish. What you can see here in these colored circles is different species that are being detected a different number of times. The bigger the circle, the more detections. So we're hearing from our fish. The network is up and running. And this network will last far longer than this project does. It will last probably a decade or more at least. This is a really exciting piece of work for the future. And by hearing from these fish, it allows us to work out where the most important bits of habitat are for them. And we need to know which bits of habitat are absolutely essential for all the various different fish. And that's because in the end, we want to get to this ecosystem-based management that I told you about at the beginning. And fisheries, of course, have traditionally been managed using quota. We limit either the number or the total mass of fish that can be caught. But increasingly now we're turning to ideas about ecosystem-based management. So rather than limiting the numbers of fish you can catch, instead perhaps we want to protect the places that fish need most for breeding, for feeding or nursery, in order to make sure that those vital functions can be maintained and we can catch fish outside of those. We're going to ask you a lot more about ecosystem-based fisheries management after lunch today. So please keep this in mind during all the presentations you hear today, because we really want to hear from you later as well. Okay, so that's a whistle-stop tour of Fish Intel, and I can't wait for us to start to hear about the data. Thank you.
Thanks, Lucy. As I said, wonderful. I shouldn't have said that. Now every speaker I'm going to have to introduce as wonderful, aren't I? So our next wonderful speaker, I'll stop there, is Mathieu Woillez. He's from Ifremer. They're the lead on the French side of the project. And he's going to tell you all about the work that they've been doing.
You are going to put your headset. I will be speaking in French now. Bonjour à tous.
So good morning to each and every one of you. My name is Mathieu Woillez. I'm a researcher at Ifremer in Brest in France. And my presentation is about the results from broad-scale telemetry arrays for tuna, sea bass, and pollock in the channel. I'm going to present again the objective of the Fish Intel project, although Lucy has already said a few words about it. So the objective of the project is to identify the most important seabed habitats. So for the three main species, bass, Atlantic bluefin tuna, and pollock. And to describe the movement characteristics and this and present all the data. I'm going to present the data that we have managed to collect over the last 2 years. So Lucy showed you all the array. So in blue, you have the Fish Intel receivers in dark. The dark dots are other receivers, especially the European Tracking Network receivers, and deployments were made with the support of professional fishers or onboard research vessels. In this map, you can see the different taggings. So the tagging areas that were made with the support again of professional and recreation fishers. So green is sea bass, tuna is blue, purple is pollock, red is crawfish, and yellow is bream. So we adopted different strategies. On the French side, we had a very large campaign in the springtime, so once a year, whereas on the UK side and the Belgian side, they conducted more smaller campaigns, but several times, but more numerous campaigns. So to tag the fish, we asked for the support of professional and recreational fishers. So they help us tag the different species. So what data have we collected? As Lucy explained, we collect acoustic data. So we have an acoustic tag that transmit a unique acoustic signals, and this signal is detected by acoustic hydrophone that decodes the transmission and adds date and time stamps. So all this data are collected and then stored in a central database hosted by our Belgian partner, Vlies, which is the Flanders Marine Institute. So all the data are put together in this central database, and we have information on all the fish and species that have been tagged. So you can see here you have the receiver identification, deployment date. You saw deployment time, deployment latitude, deployment longitude, and the detections that were made thanks to these tags. So this is an example of the kind of information and data we have obtained. So we can calculate the average number of detections per day, for example, or the average number of detected animals per day, because some animals will come only would just pass by the receiver, where other animals will stay in the area of the receivers and will be detected several times during the day. So we can see that our array of receivers is quite efficient. And we have been able to detect animal at all our sites. If we now take a closer look at detections per species, so for brass, we have been able to detect brass at all sites. So I can see the circles. The circle gives you an indication of the number. So the bigger the circle, the more animals have been detected, are detected per day. And in the blue spots identify the stations. Regarding pollock, we had detections in Cornwall and the Irish Sea. So it was more, so we can see that, and not at other stations, at other sites. So, but we have been able to, it was, and this is because it's more difficult to tag pollocks. As regards bluefin tuna, we have more detections, a lot of detections in Cornwall and northern Brittany. So we can see that tagged animals moved in flight across the channels. So this is a summary of detections per species. So these are figures that are quite different from those presented by Lucy, but these are the figures that come from the central base I presented earlier. So this includes also other data, not just fish and tail data. So for pollock, we have 83 tagged animals, 3,526 number of detections and 19 detected animals. For sea bass, we have 514 tagged animals, 1.5 million number of detections and 288 detected animals. For bluefin tuna, we have 20 tagged animals, 724 number of detections and 12 detected animals. So in total, more than over 42% of tagged animals were detected at least once by the network. Now, if we come back to the objective of the project, which is to be able to identify and characterize essential habitats for the three species, first, we have to maybe define what essential habitats are. Well, an essential habitat is an habitat that is very essential for the species for specific functions. So we identify three types of different essential habitats: spawning areas where the fish spawn and reproduce. You have a feeding area where they feed, and nursery grounds where juveniles grow. And we see during the life cycle, the fish will move from one area to the other. And as part of Fish and Tail, we started studying the link between coastal habitats and the detections we could make. So we tried to, we used two layers of data: the habitat layer, giving information about the habitats, and detections or detected animals animals. So here you have, for example, the results in the Irish Sea and Côte d'Armor coast. And you have the average number of detected animals per day for the sea bass. So if we cross this information, we could identify the habitats that were preferred by these species, by the sea bass. So clearly, you see here that sandy habitats, coarse-grained sediments, rocky habitats are preferred areas. So this is where we detect more specimens, more individuals. Well, there are some limits to the approach I've taken because it's only 2 years. So we started this approach 2 years ago. So we need to repeat this monitoring process in order to better understand and to identify essential habitats for for the different pollux. We also have to take into account that our array on French side, for example, we have the location of the arrays have been are also allow us to continue to detect the species. So we have to consolidate detection data over several years, account for sampling bias, of course, consider life stage and functions, and adapt habitat variables to species and predict essential habitats beyond arrays coverage. Because its objective is really to be able to predict essential habitats beyond our arrays coverage. As regards the movements of fish, we demonstrated that sea bass move quite far away. So, for example, we had individuals that were tagged in the Sussex that were detected in Belgium. So 5 individuals were detected in Belgian waters, whereas they were tagged in the UK. And one of the 5 individuals even returned to the UK after 18 days. We have 4 individuals detected in Devon. So clearly, we can see that have developed a fidelity to some sites, and they move across the channel. But it's true, and fish are returning to specific sites. So we're able to detect. The protocol we use in the UK is not open for the moment. So we can detect in France, on the French side, we can detect fish that come from the UK, but it's not possible in the other way around. So in the UK, they can't detect for the moment fish tagged on the French side. So I'm going to give you an example of one sea bass that was tagged in September in Belgium, and it was detected in January 2022 and the south of Cornwall, and also in January and in May 2022, and also in July in the South Devon. Another example, another sea bass tagged in Sussex and detected in Belgium in October 2022. So we see movements that are also more local. So these are individuals that have a live in coastal habitats, and in the winter they move to reefs or wrecks offshore, and we'll have to further study this movement and why they migrate during the winter offshore on these reefs and wrecks. On the French side, we also see movements from by individuals almost from west to the east, and maybe it's the start of a migration towards spawning areas. As regards pollock, we see no movement between France, Belgium, and UK, but we see movement at a local scale. So we have strong residency of pollux in the Aras Sea, and especially near the Ushant Island. So to conclude, we also have the third species, bluefin tuna. So here you see in yellow, you have bluefin tuna detections, and when you have so the blue dots are the receptors, the receivers that have detected bluefin tuna, and the white dots are receivers that did not detect bluefin tuna. And you also have this information, but these individuals were detected in other countries, in Portugal and Spain. So most of these individuals that were stayed in the along the UK shores. Some went south to Spain. And in the winter, beginning of spring, we have very few detections. We had one detection in Spain. And in July 2022, these individuals were detected in Portugal, then France, and back in the UK. So clearly individuals that were, so these individuals that were attacked in the UK returned the next season to visit the English coasts. So to conclude this presentation, we can say that we have an operational acoustic telemetry network that is that can be applied to other species. This network has allowed us to give us new insights on fish spatial ecology. So this is really a new type of data that can be used for an ecosystem-based management, and practically can contribute to marine spatial planning. That is, finding a balance in space between anthropic activities and a healthy ecosystem. This data can also be used to identify stock in more form. So to have a regular stock assessment. But all this requires continuing tracking over many years to acquire a good knowledge of fish essential habitats and movements. But of course, this is key knowledge to protect these areas and sustainably exploit fish resources. Thank you for attention.
Thank you very much, Mathieu. So our next two speakers today are from the fisheries regulators from both sides of the channel. So our first one, we're going to start at home. It's James Stewart from the Devon and Severn IFCA.
Thank you. Yeah, so I'm here representing Devon and Severn Inshore Fisheries and Conservation Authority. I've been asked to talk to you today about how Fish Intel's research is relevant to our work in managing inshore fisheries in England. Because of a lot of what I'll say is really from an IFCA perspective, I'll just give you a quick introduction to what that is in case you're not too familiar. So we're the statutory managers of inshore fisheries formed by the Marine and Coastal Access Act, and we manage fisheries from baselines out to 6 miles out at sea. Now there are 10 IFCAs around England, which you can see on the map here. Our district is the purple one in the southwest, Devon and Severn. So we cover the south and the north coasts of Devon and the areas bordering the Severn up into Gloucestershire. So along with the other 9 IFCAs, we have a shared vision, and that's to lead, champion and manage a sustainable marine environment and inshore fisheries. And to achieve that really by achieving a balance between the social, economic and environmental aspect of exploiting sea fisheries. And really there are 3 aspects of our work that I'd like to talk to you about today, where evidence like that collected by Fish Intel can feed into what we do. And the first is initially just direct management of fisheries activities. As many of you will know, that's managed by a combination of national legislation along with local bylaws. So in our district, for example, we have a range of activity-based bylaws which manage mobile fishing, netting, diving, and potting. And then there are also voluntary measures that go alongside those. So these all regulate things like acceptable gear types, catches and landings that are acceptable, and also restrict fishing activity over space and time. These all at different intervals will have to be reviewed, and we review those based on the best available evidence at the time. And that can involve things like evidence on the direct impact of the fishing activity on the target species. But also we need to know how effective our management measures are actually being, whether we're achieving the goals we set out to. And I'd like to talk about that in the context of Fish Intel's work on bass at first. So we all know it's a high value species, both commercial and recreational fishers, but unfortunately it's experienced high declines in spawning stock biomass recently. And to attempt to manage that, there's been a lot of kind of restrictions on landings, which can lead to high levels of discards in some fisheries. The thing is, we don't always understand how well these fish survive the capture and discard process. So we've set out to try and address that at small scale inshore fisheries by partnering with the Fish Intel team in Plymouth. So working with the team, we partnered with a local fisherman in Salcombe, which isn't too far from here, try and understand how well fish caught in gill nets will survive when they're discarded. So we were working from a small boat and then transferring fish onto this larger boat where we had a kind of an operating theatre essentially, where the team worked on these bass. And over the last 2 years, so January this year and January 2022, a total of I think 86 bass were tagged. These are tagged with the acoustic tags, which you've heard about already this morning. And in partnering with Fish Intel in this way, we understand not only how well fish survive immediately at the vessel after they've been captured, but also by looking at their detections in this acoustic receiver array in Salcombe and further along the coast, we can understand how well they survive once they've returned to sea. As I say, that's really important for us understanding kind of the effectiveness of broader scale measures to do with discarding, but can also inform management at a regional scale when bylaws are reviewed and that kind of thing. So the second element I'd like to talk about is the our management of fisheries activities in marine protected areas. And really, I'm going to have to go into kind of not too much detail. I have to be quite broad brush because I'm trying to cover quite a lot. But this map shows the 22 marine protected areas in our district, or that are partly within our district. You can see there's a lot of them, and across these 22 marine protected areas, we've conducted assessments to look at what potential impact fishing activity can have on the designated features. For those assessments, we use whatever evidence is available. A lot of it will be based on our own research, literature reviews, including a lot of the work that Fish Intel team members have produced. Where we do those assessments, and it looks like we need to introduce new management, we can introduce management such as we have in the Severn Estuary SAC, for example, and the Tall Bay and Lyme Bay SAC, where these designated features, so seabed habitats in this case, are protected completely from demersal towed gear. So that's an example from our mobile fishing permit bylaw. This is an example from Lundy, where potting restrictions are in place to support the no-take zone around Lundy. And all of these are based on evidence of the impact of fishing activities on those designated features. The third element is marine development. So we're regular or statutory consultees on a range of marine developments in our district. That can range from things like tidal power, nuclear power, to aggregate dredging, port developments, and mariculture. And our role as consultees when we're responding to this kind of thing is to try and ensure that developers lessen, mitigate, or compensate for their impact on habitats, fish, and the fisheries that depend on them. Mathieu and Lucy have already talked about some of the evidence that's been gathered. If we can identify where fish are, what their habitat associations are at particular life stages, and how they use those. That can really feed into that kind of marine spatial planning process and help us to, as I say, lessen or mitigate the impact on fish. And that's obviously going to have benefits, not only for the fish, but also for the small scale fisheries that we try to manage. So the final element that I'd like to talk about just quickly is our fisheries research and management plans. Now, these are distinct from the fisheries management plans that you will have heard about probably at a national level. These are a project we've been working on for a couple of years now. And basically what they are is a review of the ecology of certain fish species. They go into the different fisheries that depend on those fish and the management measures that apply to those fisheries. Alongside that, they've got an element of a review of the other impact that the fisheries can have, for example, on habitat, but also on the other pressures that the fish species face beyond just fishing. So it's, in that sense, another step towards this ecosystem-based management. A lot of this is based on literature reviews and some fairly extensive stakeholder engagement. And I'm pleased to say that the team at Plymouth in Fish Intel have really been instrumental in leading our stakeholder engagement for one of our recent bass FRMP. Now, through this process, we've identified a range of key evidence gaps that research such as that formed by Fish Intel can fill, and come up with recommendations for management, future fisheries management in England. And what we hope this can do is not only be useful for us in engaging with stakeholders and encouraging new research through projects like Fish Intel, but also to help the implementation of the national fisheries management plans, which are being rolled out as part of the Fisheries Act, under the Fisheries Act. Now, that's something I had hoped to talk about in a bit more detail, including the fisheries objectives. But I think I'm swiftly running out of time, so I'll leave that potentially for questions or in the break. And yeah, thank you for having me. Sure.
Thanks very much, James. We've also got Libby from Natural England speaking after the break, so hopefully she'll cover some more on the last year fisheries management plans. Where are you, Libby? You made it, good. Okay, thank you. So now we're going to turn to the other side of the channel. So if you don't speak French, we need your headsets. And I'd like to introduce Erwan de Meneur, and he is from the French Sea Fish Committee's equivalent. Thank you, Erwan.
Hello, everyone. My name is Erwan Quemenuer, and I work with the Finistère Fisheries and Marine Farming Committee. Really speaking only from the point of view of professional fishermen. It's the professional fishermen themselves in France who manage the fisheries. Fish Intel is an innovative project which brings together all players and stakeholders. We at last have an opportunity to track the fish and crustaceans using telemetry, and this is going to provide us with some major information. So we're going to talk about pollock and bass, sea bass. And over the past 5 years, were many restrictions that were placed on sea bass. And the professionals are really raising the alarm to say that the stocks are maybe not going to be sufficient, that the yields are much lower. Is this due to fishing pressure? Is this due to a dwindling of the resource? We don't know yet. So sustainable fisheries has been the key approach of French fishermen for several years now. The French fisheries have well understood that trying to obtain MSY as quickly as possible on the largest number of species possible would be a way of mitigating fisheries pressure, and the boats have the right to work in a sector with a given type of gear, targeting a specific species, and in these specific areas, the idea is to avoid concentrations that are too important, and we have octopuses that in fact coming into our area, and we're trying to avoid these concentrations insofar as possible. Fish and Tail enables us to obtain a maximum amount of data. As I said, if you really want to manage a given species, you have to know that species well. And if we close an area, if we have a restricted access, a typical example in the Bay of Biscay is the fox zone that was in fact closed to fisheries, and this is the case in the Bay of Biscay and elsewhere, and what that leads to is discharges and prohibiting the fisheries for hake. So the idea is to understand these habitats and to see what we have to do. Should we forbid below a certain size or in a certain area? But trying to create a sanctuary, really a protected area, totally is not always easy or possible. If we try and provide restrictions for a given species, we have lobster, for example, on the French coast that have become an endemic species. Every time we try and apply global or general protection measures, in fact, we're going to have an impact on the entire ecosystem, and a certain imbalance that could take place, could come into being. And we have to take account as well the social and economic balance as well. And these, if we have a moratorium on sea bass, when we had the moratorium on sea bass, didn't that create an imbalance for other fisheries as well? And is that really what we expect from fisheries management? The French fishermen are not convinced of this. So for us, what is the best management technique to be applied? Well, in France, we feel that it's important for the professionals to, in fact, take on board and buy into the results from the various scientific programs and projects. Let me give you the example of crawfish, for example. We had strictly regulations that applied strictly to the professionals for a closure zone. And we had an increase of the size from 110 millimeters instead of 95 millimeters. That was voted in France. Then we had a closure of fisheries, no fishing of this species in January, February, and March. And then we had the prohibition of fishing females that were egg-bearing, that had eggs, and of course, to better manage this and to fight fraud. This is what we've expected for the past 10 years. We're expecting the stock to rebuild. And we had some a dozen tons maybe of production in 2018, and it went to 120 tons in 2022. And this is a reflection of what the professionals were able to implement because how should I say this? This shows the interest of, in fact, of creating these measures and closures, and the professionals saw with their own eyes how interesting this was. So this means we can give professional fisheries organizations the elements that enable them to bear out or contradict, in fact, what their empirical knowledge has provided. For example, you have some fishermen who are going to tell us that the sea bass stock, in fact, in the Erwan Sea, much further north in the Bay of Biscay proper. And you have the fact that the adult crawfish are much more sedentary than the young ones. And Fish and Tail enabled us to show that what seemed to be impossible 5 or 6 years ago, when the tagging of pollock seemed impossible, it has become so. And now we're going to have to develop, in fact, working with the professionals, this network of acoustic receivers and the tagging, and that what will remain is to tag the maximum number of individuals, and why not other species, so that we have data about their behavior. Thank you for your attention.
Thanks so much, Erwan. That was brilliant, and we really, I can hear my own voice. We really appreciate having the fisheries and regulators involved with this project. It's so important that all this research gets to where it needs to get to as quick as possible. So I don't envy your job. I'm really pleased to be working this side of the collecting science side of things. So we do have now 15 minutes to ask our speakers questions. So can I ask the speakers from this morning to come up? And we just need to be around the microphone when we're answering the questions so the people at home can hear us. We have microphones. Alice? Ada? Brilliant. Okay, so is there anyone here in the audience who would like to kick off the questions? Oh, at the back. Hi, Randy.
Very good morning. All right, brilliant.
If you'd like to say who you are and where you're from, that might help.
Randolph Eltrop from Natural England, so from the UK. A fascinating series of talks. And I was wondering, I had a couple of questions for Mathieu on the bass tagging. Could we go back to any of the slides at all, Emma? Or
Alistair, is that possible to go back to Mathieu's talk?
The long range slides are really fascinating to see that there was a lot of site fidelity with bass coming back to the same sites, even though they do long range migrations. But you mentioned on the slide that there weren't any cross-channel movement detected. And I was kind of wondering if they were moving between English and Belgian waters, but not English to French waters. So I was wondering kind of what the rationale for that might be.
I have no rationale about why they're moving. Why we detected the rather movement toward the.
Yeah, so I guess they do long range movements east to west to Belgium, but none went north to south.
Well, it's conditional to the tagging, to the individual that you tagged, I would say first, because you may tag a specific cohort that have a specific migration movement. So for the moment, I mean, this is one few individual doing this. I would rather wait for a consolidation of those data to extract like a robust pattern. However, we have been working on another project in France. We have been tagging 1,220 sea bass from Dunkerque to Cabreton in the south of Bay of Biscay, and we have been. So it was on the French side, and we were able to see pattern for those fish from the Bay of Seine and Dunkerque that stay in the north and moving westward to spawn. And we didn't use acoustic telemetry, but rather geolocation modeling, which is a slightly different way to track fish. And fish that were in the northern Brittany, they were mostly moving in wintertime to spawn in the Bay of Biscay. It's like you got two identities moving like that. And we did also genetic analysis, and genetic analysis was showing like a discontinuity in the exchange, in the demographic exchange that you have between those two units. So right now, those knowledge that have been produced by another project actually that are pushed to ICs to revise stock identity for sea bass. So why we did acoustic telemetry tracking was for us to get observation data using acoustic detection to confirm what we have been able to see with a modeling approach, where you only have the release position of your fish and then the recapture of your fish, and then you infer the trajectory just by comparing sensor data, temperature data that have been recorded by the fish. We are using archival tag, and we compare them with sea surface temperature, for instance, and you reconstruct the possible track. But with acoustic telemetry, then you will know at certain time date where your fish was present. So then the trajectory that you infer is confirmed by observation. That's why we wanted to use sea bass as a case study in this project, and because we knew that we have the knowledge to tag such a species, it was easy. So the challenge for us was to put receiver at sea, because we were not experienced like our English colleague, and our focus was mostly on pollock, because what Erwan say, there is a big gap of knowledge about the ecology of pollock, and we wanted to work mostly on this species.
So the bass tagging on the French side was done using data storage tags, and the English tagging was done using acoustic tags.
For fish and tail, we use acoustic tag too.
Ah, okay. But still no movements, only movements east to west. So they do the same on the English side and the French side, but they don't go north to south.
For the moment, no, we didn't see any of this.
So Brexit borders. Okay, thank you, Saturn.
But as I said, I mean, right now there is a compatibility issue. We are able to detect sea bass that would move to the French coast, but let's say the sea bass that have been tagged with our acoustic tag cannot be detected on the receiver from the UK. It's about a protocol of communication.
Ah, right. So it's it will
be updated pretty soon.
Okay. So they're not compatible. The receivers and tags are different frequencies. Right. Okay. That makes sense.
You have to be aware of it.
Okay.
And just that you know, when you put an acoustic tag on a fish, we are using tags that last for 5 years. So we will have, the idea is to get 5 years of monitoring, and then you will get robust pattern of movement.
So is it to do with tag protocol, so the open protocols, or is it different frequencies? So is one 69 kilohertz and one 180, or is it that
we are both using 69 kilohertz frequency?
But it's the tags that aren't compatible.
Yeah, so we are using the open protocol recommended by the European ETN. And then there is Innovacy that is using a closed protocol, but that will move, normally would move toward the open protocol in the coming autumn.
Brilliant, thank you.
Yeah. And I think the Fish Intel project and all the consortium, I mean, we work hard to make it happen, and that's really a way forward.
Robin Bradley from the Bass Angler Sport Fishing Society. Question for James. I'm very interested to know what they found with the bass that were caught in gill nets, that were then tagged and released. How many of those survived and for how long?
Yeah, so there's a few elements to that answer. The first is that I'll be reporting on that to our authority, which is basically the committee that runs the IFCA in 2 weeks' time. So we'll be able to release a lot more of the information then. The data that the Fish Intel team have, which are on the bass that were actually tagged, I think last year 49 were tagged, and we can be sure of the survival, I think, of about 95% of those at least beyond a few months. So that's very good. Not all of the fish that were caught were tagged. So that's the element of the data that I'm working on at the moment. I wouldn't be able to recall off the top of my head exactly what the mortality was, and I think it would be unfair to give the picture just of that, because obviously you've got to take into account what proportion of the catch was bass bycatch to sort of take in to really understand what the impact of the gill nets was. But yeah, we can share that very soon. Yeah, sorry that I can't do it just now.
Hi, Jean-Luc Solant from Marine Conservation Society. A question really for Mathieu. I didn't get to ask it yesterday at our workshop. The data you suggest is that the bass and the Legion, the pollock, are in more sedimentary areas. Is that possibly because there are more receivers in sedimentary areas, or do you think that's a true reflection of the habitat use of those fish?
No, I think it's bias.
Thank you.
I think it's bias, because when you put a receiver close to a reef, I mean, you in the detection range of the receiver, it's like a radius of 700 or 500 meters around the receiver. Right now, in the computation we do is you account for the area of each sediment. So you may have more sand around the reef or the wreck in comparison. So then there is more in the habitat use. You have more sand detection that are allocated to sand habitat, but then I don't think it's a real. I mean, the presence of the reef or the wreck is more important, even though the area is smaller. Maybe smaller. So yeah. But if you would, the thing is that I think the way we deploy the receiver is biased to optimize the tracking of the fish we want to follow. But if we have poor interest to put a receiver in a sandy place where there is not much expectation to see a bass. So, I mean, these are expensive, so you don't want to track the fish you tag. So, but so you have to account for those bias.
I'll just add as well, we'll be hearing from Pete later, who'll be talking about using this equipment for the fine scale telemetry arrays, where you can actually pinpoint species to a meter and therefore get better habitat information that you're asking about, Jean-Luc. Okay, I'm just going to turn to Slido and ask if there's any questions from the people at home or on Slido.
Yeah, we've got quite a few questions online. So the first one is for Mathieu again. If you want to step up to the microphone again, Mathieu. So this one is just kind of a clarification question, I think. And I think you covered it when you answered Randy's question, but it's just from David Curtis. And he says, sorry, I didn't understand the translation. Are sea bass crossing the channel from the UK to France and vice versa? Or is there a technical issue, meaning this cannot be measured? I think
So
yeah, okay. I can't get any feedback from David, so I'm not sure how he feels about that. There's another question from Jenna Edwards, also from Mathieu, and she asks, what company manufactured the tags used in your study?
So we go for the whole consortium. We have two providers, so Innovacy and Telma Biotel.
Great, thank you. See if there's any more questions for you, Mathieu. Okay, yes, I've got one. Got one for James, another one for James. So this is an anonymous question. And it says, I would be very grateful to learn more about the study IFCA carried out into sea bass discard survival for 4-inch thick nets. What was the outcome?
Yeah, so I think that's also been addressed, and we'll be sharing that report as soon as I can.
Did you want to ask the one in French for Erwan? Question for Erwan.
Are they a species of fish that move in an unexpected way? Did you have really big surprises in the results you obtained? No, not really, because for crawfish, we had also empirical knowledge from fishermen, and so the movements were not unexpected. And as regards the sites, we had no information. We know that these fish species are quite sedentary, but we don't know, nothing unexpected.
Thank you. Any other questions from the room? Yeah.
Hello, so I'm Emily from the Marine Conservation Society. I've got a social science question, and it's for someone in England, but I'm not sure who's best to address it. But I was interested to see that on the French side, there was a lot of inclusion of fishermen in the process, and I wondered if there'd been anything on the UK side, whether it was involvement in tagging or actually community consultation itself.
Yeah, I mean, the expert is smiling at me at the moment from the back of the room. And you are going to hear a lot more about them from their talks as well. So if you could wait till after the break, you'll hear from them. And then we've got an hour workshop with them as well this afternoon. So come on then.
Yeah, so we cannot catch and tag Atlantic bluefin tuna without support of the recreational fishing industry. So every bluefin we've ever caught has been a partnership with recreational fishers. And there's now a scientific-led recreational fishery that's opened, just finished its second year last year to catch bluefin tuna in the UK. And that's, I think, a great product that goes to show like how much we need each other to do good stuff. So yeah, they're integral to all of us are, you know, rely on the fishing industry to make a success of the science we want to do. They're very valued partners.
Any other questions from the room? No. I think everyone's ready for a coffee. But okay, thank you very much to all our speakers this morning. It might be worth me just clarifying this issue we highlighted about this lack of communication between French and English tags across the channel. And one of the most important ambitions for the Fish Intel project was to resolve this issue of different companies having different technology that don't speak to each other. And so this was the key thing that we really wanted to sort out. We want to make sure that every single receiver can hear every single fish. And this has finally been achieved. And this through the weight of all these project partners and the buying power of multiple countries and organizations. So we've got the biggest kind of seller of this type of equipment has finally agreed to join this open protocol we're talking about. And so we are now at a point where this software patch will be applied to all of our kit, which will make sure that all the tags and receivers will be fully cross-compatible by October 2023. So it's definitely one of the good, the best achievements of the project, I think. So I'm going to let you go and have a coffee and a snack. I think it's been served in the foyer. And I'd like you back here for 10:55, please. Okay, all right. I hope that everyone's refreshed and ready to go again. So our next session, again, has a mixture of fish intel and external speakers. Our first talk is actually going to be shared between Tom Stamp and not Mathieu, Marshall. Marshall, are you going to speak in English? You're going to make it difficult, aren't you? Lovely, lovely. Okay, so we'll start in English with Tom Stamp, and then we will switch to French with Mathieu from Ifremer. Mathieu, Marshall, sorry, from Ifremer. Okay, Tom, save me.
Yeah, thanks everyone for coming today. So my name's Tom. I work under Emma at the university, and with Marshall, we've been tracking crawfish in the UK.
Donc Marcel Laurans, moi je travaille à Ifremer à Brest.
My name's Mathieu Laurens. I work at Ifremer in Brest.
So just a little bit of context as well. So these are crawfish. These are otherwise known as spiny lobsters. Used to get them quite a lot across most of the coastline around sort of southwest UK and northern France. But I think it was in the 70s, was it, where there was a big population crash. And quite recently, this population has rapidly expanded, and that's led to an increased interest in fishing pressure on the species. And so working with the Scilly IFCA and Comité de Pêches in northern France, we've been tracking these animals to look at is spatial management an appropriate tool to conserve these species. So in the UK, we've been doing a lot of work in the Isles of Scilly, where there's a number of protected sites known as marine conservation zones, but dotted around the islands. So these are the islands here. And in each of these sites, crawfish are sort of a designated feature, and so you're not meant to be doing activity within these sites which could potentially damage the population. And then Marshall is just going to give you a quick brief on a similar site which is in northern France.
Alors, tout à l'heure, Erwan du Comité des Pêches vous a parlé
Earlier, Erwan from the Fisheries Committee spoke to you about the regulations that were put into place for crawfish in France. And he talked about the closure of the best fishing area that was actually here. The specificity of this zone was that no fishing of crawfish was possible. It was closed in 2007, and as of 2009, we started monitoring abundance in this zone.
Okay, so over the past 2 years, we've been tagging crawfish with these acoustic transmitters. And so unlike the fish, where we put the tag inside the animal, with the crawfish, we glue this to the carapace of the animals here. And over the past 2 years, we tagged 120 different individuals across both the French and the UK sites. These cover a sort of broad size range of them and the different sectors. Interesting to note as well, we'll go into this in a bit more detail in a second. We've only managed to tag 21% of the animals we tagged have been female, and the remainder have been male. And we'll go into why that might be the case quite shortly.
Et on peut aussi compléter.
We can also complement that by seeing what we observe here, that this is noted by all fishermen, in fact, and everyone's asking why there are more males than females.
Of type of receiver array that we've deployed on the Isles of Scilly. So each crosshair here on the map is where we deployed our receivers. And these sort of generally cover an area of around 2 kilometers across a sort of rocky reef system off the back of an island called St. Martin's. And while we haven't got a map of the French equivalent, the receivers were set up in a very similar way in there, so we can directly compare the results to 2 sites. Okay, so this is quite a boring graph, I admit, but basically, so this is called an abacus plots, and it basically just shows when our tagged animals are present or absent from our particular receiver array. So basically, each line represents an individual animal that we've tagged, and you've got time along the bottom here. And so the plots here on the left-hand side are from the French sites, and so these are the females, and then these are the males that have been tagged, and then these are from the Isles of Scilly. Basically across the whole study, we've redetected our sort of tagged animals 375,000 times. So it's a huge amount of data to get from a study like this. And one of the key things that we noticed is that we get a lot less detections from the females than we do the males. And I don't know whether if you want to elaborate further.
Okay. Et donc, en complément de ce que vient d'indiquer Tom.
To supplement what Thomas just said, for the French side, the crawfish we marked was a place where we're monitoring experimental fisheries and have been doing so for several years. And what we wondered what the crawfish did in wintertime, and those that are visible here over 5 months don't seem to have moved from the area where they were caught and the area and marked and put back. So we see a degree of sedimentarity, a fairly high degree of sedimentarity for a number of them. And again, this difference between females and males.
We are picking them up, moving around between different receivers. So the color of these points is the different receivers, but fundamentally these animals are staying within the receiver array. So that means that they're staying within an area of around 1 to 2 kilometers, and they're not really moving beyond that. And that's quite critical when we're talking about spatial management. A really key development that we've also managed to get is some of these animals we've attached depth transmitters. So the tag will go off constantly. And then while it's emitting the ID of the animal, we also get the depth that animal's at. And so this is a 3D model of the site in the Isles of Scilly that we've been monitoring. And the green is the areas where we've managed to sort of isolate where these animals are hanging out. And so the animals that we've tagged in the Scilly seem to be hanging out a lot on these sort of reef tops here, which are around sort of 20 meters, around the sort of mid bit of these sort of marine cliffs. And we haven't really picked up many of the movement down to the bottom, which gets to around sort of 80 meters at the bottom. And then
nous avons pour l'équipe française, on va faire le même travail que celui que nous avons.
We did the same thing that Tom's team did. The big difference between the two zones, even though the habitats are similar, is that in the French areas we have much stronger currents. And so the crawfish go lower and deeper in wintertime to protect themselves from the tide and from the currents. And so here you see the depths. I would like to emphasize the fact that these crawfish that we've been monitoring since 2007, 2009, that are caught each year to measure them, all those that are caught are tagged with a spaghetti tag. And the results that we have are that every year in the same place, we recatch a number of our crawfish. We see how much they have grown. And the one here was first tagged and caught in 2011, and 8 times it was caught and identified in the same area, but we didn't know what it was doing in wintertime. And thanks to Fish Intel, the acoustic aspect gave us more than just by just tagging an individual. We have between 2011 and 2022, it seems that the animal stays at the same place in wintertime. And this really gives information, as Erwan said earlier, to the fishermen, that the Fish Intel acoustic approach provides information that previously fishermen suspected but could not confirm. And so this means the spatial management of fishing activities is a very important element.
Okay, so yeah, just to sum up, basically, we still have a lot of data to collect, but the sort of early results that we've got from the study show that sort of males in particular are definitely hanging around a really quite small area, and they don't move much beyond that over quite long periods, over about a year. And that seems to suggest that, at least for the males, protecting an area of seabed would be an effective way of conserving these animals in the future. We definitely do need to collect more data and see how it varies across space and time, but the early results have been really great for this project and really help inform management at both the Isles of Scilly and in northern France. And so, yeah, thank you very much. Yeah.
Thank you very much, guys. That worked fine. That was good. If anyone has any more questions about that project, we've got two IFCA officers from the Isles of Scilly with us today, Tom and Ricky. So they're over there. Okay, next up is another homegrown Pete Davies. There he is. He's hiding. And he's going to be talking more about the fine-scale telemetry tracking at Fort Blackberry.
Everyone, wonderful to see so many of you here. Old faces new. I'm going to be talking a little bit about our work up in Sussex, where we've been looking to address some of the uncertainties around the reproductive ecology and the movements of this wonderful charismatic species, the black bream. But just zooming out a moment, I thought I'd just talk a bit about spawning aggregations and what they are. So many of our most popular fish for recreational and commercial use gets together at certain times of year at certain places to aggregate to spawn. And this presents a bit of a challenge for fisheries management because, as I mentioned, these spawning aggregations are often predictable in time and space, which can make them a bit vulnerable to overfishing, overexploitation. The proper management of these aggregations to ensure sort of sustainability of these fish species requires us to know sort of the places they're getting together to spawn, how they're behaving during this reproductive period, in order to come up with ways of managing them. But for many species, this sort of information is lacking, including black bream. So up in Sussex, at the beginning of the of last year, we set out to address some of these questions with regards to black bream in an area called the Keynote Rocks Marine Conservation Zone. If you're not familiar, the Keynote Rocks is a rocky reef just to the south of Littlehampton in Sussex. And every year it's becomes a mecca for black bream fishermen who tend to come there to angle recreationally black bream. And in terms of the management of the site, this is the shape of the MTZ, and you can see it's got various zones here, four zones with different management. Zone 1 at the top there is a complete no-take zone for black bream during the nesting season. Zone 2 encompassing quite a large part of the Keynote reef itself. You're allowed to fish, but it's a bag limit is imposed during the spawning season. And in terms of management of black bream, one of the main questions surrounding their proper management is what happens when you catch a black bream and release it. How does it affect their behavior going forward? Because in terms of their ecology, they're quite an interesting species in that the males will dig out a nest in the sand, and the females will lay eggs. The males then guard those eggs for a couple of weeks before those eggs hatch. So kind of one of the key questions that managers are quite interested with regards to black bream is what happens when you remove a nesting male. How does it and then subsequently release it? How does it affect its behavior and its ability to guard those eggs? And so to try and address some of these questions regarding sort of black bream ecology at the Keynote, we set up a fine scale telemetry array. The most of what we've been talking about so far has concerned the broad scale movements of fish, so long distance movements and their presence or absence at certain telemetry receivers. In this case, on the Keynote, we set up a fine scale array. So this is where you set up a cluster of receivers in quite a dense arrangement, and it means that any fish moving through this or indeed nesting within this area, the signals from the tags can be picked up by hopefully multiple of these receivers, and by calculating the slight difference in time of arrival of the transmissions of the tag at the different receivers, you can work out, hopefully with sort of sub-meter accuracy, exactly where that fish is on the reef at any given time. So I hear you asking, what does living the bream look like? So you start here on the deck of, this is Watchful, the patrol vessel of Sussex IFCA, and there are all our receivers laid out ready for deployment. And once you've deployed them, you deploy your expert fishermen, try and catch the bream within the area. You then wait, sometimes for quite a while, before you catch some fish, tag them with an acoustic transmitter, and release them back into the water. Once you've done that, you're really happy because you've got rid of all your tags. And then the hard bit, retrieving the receivers, downloading the data, getting quite slimy, but hopefully coming up with some good results. So another boring graph, but thankfully Tom's done a good job of explaining what an abacus plot is. So each of these rows represents the detections of individual fish. And these are 25 black bream that we released within that Keynote rocks fine scale array. And there are several things to draw out here. So this dotted line here on the left, that's the point at which we released them in early May. And this second dotted line is a second release of only 3 fish. As it turned out, it was quite hard to catch them at that time in mid-June. And then this solid line is the time we downloaded those receivers. So you can see here that the vast majority of the detections of those fish happened within the first 2 weeks after release. And then kind of a conspicuous absence basically from that Keynote site. So kind of suggests that the residency of those fish during the spawning season was quite transient. They were there for about 2 weeks and then they left. And very few of these fish were detected on our wider array that we have deployed in Sussex. So the question of their sort of habitat use outside the spawning season is somewhat of a mystery, and something we're going to hope to try and address going forward. But if you zoom in on those individual detection histories, you can see that for some of the individuals, these solid looking bars, we're getting really good, strong patterns of detections within that fine scale array. For others, it's a bit more patchy. And interestingly, when you overlay the hours of darkness on this abacus plot, you can see that for a lot of those individuals, they seem to be more likely to be detected during the day than at night. And this is drawn out, this is reflected a bit in these sort of wagon wheel plots, if you like. The one on the left is the, if you imagine that's a wheel showing the 24 hours in the day, the clock. And during the day, you tend to see more fish within that array than at night. And likewise, on the right, you've got the number of detections. There tends to be a stronger pattern of detections during the hours of daylight. And for three individuals who managed to perform this fine scale tracking process. So this is an animation, I think about 10 days, the life of 3 bream, and it cycles between day and night. And there are a few things to kind of pick out here. The first, which you could hopefully see, is that at night, fish tend to be a lot less active. So sort of roaming onto the sand during the day sometimes, and then quite a bit less active at night. Secondly, each of these individuals tends to return to the exact same spot on the reef during the hours of darkness. It's as though they have these particular areas on the reef, almost like a roost, which each individual will return to at night. There's some really interesting sort of behavior and ecology that we're starting to pick out with these bream. Unfortunately, these 3 individuals that we managed to do this fine scale tracking process on, when we tagged them, they were all female. So it's unlikely that this behavior represents the nesting behavior that we were so keen to try and dig into. But fortunately, as Lucy mentioned earlier, female black bream turned male. So hopefully next year, when we come back and repeat this process, we might be able to get a bit further with that one. And this, thanks, Sheehan. This wagon wheel demonstrates simply what I was talking about earlier. There's a strong diurnal pattern of activity with black bream. And if you speak to anglers, they'll tell you that they tend to be a lot easier to catch during the day and a lot harder at night. And this kind of illustrates that they tend to be more active during the day out and foraging, whereas at night they tend to be hunkered down, probably roosting and sheltering from predators. So in terms of our next steps, and beyond fish intel, we're massively keen to keep this work going forward. The tags that we put in the black bream that we release onto the reef will last 3 years. So hopefully come this spring, we'll be back out on the Kingmere, repeating this process and hopefully capturing the behavior of fish that return to the site. And that's a massively important question in terms of the management of the site. You know, how loyal are black bream to particular spawning areas? Do they show homing or fidelity to certain sites? But we ticked off a few things. We found out that it's possible to track bream using this fine scale method. And we have certainly highlighted some interesting aspects of their ecology, and we're really keen to keep this work going forward. It was a massive collaborative effort, and so it's wonderful to see some representatives from Sussex IFCA in the room today. We couldn't have done any of this work without them and their expertise on the boats, as well as Natural England, who funding the purchase of acoustic tags and receivers, alongside Fish Intel, made this work possible, as well as the Sussex Wildlife Trust. So thank you very much, everyone, for listening.
Thank you very much, Pete. Okay, as I mentioned, we've got the Scilly IFCA in the room, and Tom Hooper is up next. They are one of the Fish Intel project partners, and Tom will be talking about how we connect fisheries and conservation management for species that move.
Thanks. Yeah, I'm from the Scilly IFCA. We're part of the same family that James was talking about earlier. We're just smaller. There's both of us here today. So we have responsibility for management of crawfish as a conservation feature, but also as a fishery. In other words, we have to strike that balance between its commercial use, but also it's the kind of conservation value that it has. So crawfish they're unique. They are both a feature, a conservation feature that has to be protected, and they are within, as others have shown, within 9 of our 11 zones. Just to be clear, feature it's a technical term, which means that they've been part of lists. I mean, in some cases going back to 1979, which means that they had to be included, I think, in the network. And you can see here in the map on the right, I'll come back to this in a second, they're both showing in the southwest 67 sites with crawfish in them, and then those within the Isles of Scilly. What I really wanted to do was try and connect these two aspects here. They're almost seen as separate, and wouldn't it be great if we could connect them together and ensure that research from fishing, research to conservation. So you could, I can't. I wish I could. So if I refer to the map, the column on the right. Okay, I can do. The column on the right is, and I can see it here too, is concerned with the almost the conservation aspects. Column on the left is aspects to do with fisheries, and you can see they're very different. The data informing management as a fish is different from the data informing management of crawfish as a feature. And as I say here, wouldn't it be good? The two really need to complement and inform each other. And it's, the question from me to you really is how we can do that better. And I think this part of the workshops, I didn't know this, but I think it's really sort of feeding in and kicking you off into the the workshop we've got this afternoon. So just the map on the right is it's a bit hypnotic, but if you just look at it for a second, I'll explain it. It's from Catherine Whitley's work in 2021. The regular rectangles show the abundance of crawfish, pale yellow to red increasingly, and then the kind of bigger pink polygons are the marine conservation zones. It's worth remembering that when these were designed in 2011, crawfish were fairly rare, and crawfish were not a strong influence. In fact, they're a very weak influence on the design of these MCZs. So it's almost a bit of an accident that there is overlap. But of course, they kind of future-proof and will make sure that they are protected into the future. That's not mine. Okay, that's mine. So underpinning the conservation is sightings data, and sightings data is primarily made up of diver observations, and there's great work from Angus Jackson published 2022. He collected all together all of the diver sightings data, and it's a 1 kilometer resolution, and he used an occupancy model which basically said, well, here's crawfish and here's where crawfish are probably going to be. In other words, it smooths out that skew, that diver, where divers go and where they might have gone, means it's smoothed out, and you get a pretty accurate understanding of where the crawfish are. And you can see on the left there, that's 2000 and then 2020. So they're increasing in number. They're going a little bit further east and they're going a bit further north. So that's really what's underpinned the conservation. The question is, well, how does that correlate? So remember this, and you can see there, on the chart on the bottom, the increase in numbers that we've seen over the last 15 years. Then in terms of and Tom's talked about this, but I want you to see this all in context. So do the crawfish hang around within the MCZs? Yeah, great. So we've got that confirmation, but there's still a lot of uncertainty about how, where they're going. And of course, they are going outside the network sometimes, but it seems that there's a large population staying central within the protected area network. So if you then go on to the fisheries data, we've got two things that we can rely on. There's the general landings data, which show a big picture, and it's really the map on the top, the chart on the top right shows that it's quite a familiar graph now, isn't it, from 1920s, a little bit of an upsurge through 1970s, and then a decline over the last, really up until about 2014. Then on the left, top left, you can see there's been this incredible increase of crawfish locally with us, and we can see there's been a big increase in the crawfish, which is fantastic. But then the question is, how sustainable is that? Are the marine conservation zones doing their job? And our job, of course, is to make sure that that sustainability is maintained. And of course, and this is the great point, we don't speak to each other across the channel. Is that the same picture in France? And I think that connectivity is really, really important. And likewise, so this is something that I really challenge us to be able to think about later on. So we know then we've done our own research with fishermen and this great collaboration, Mark and us here. We've been on board his boat and 5 or 6 others. So we're measuring, sexing, and we are positioning every crawfish that we catch. And this is work that Sarah Morecambe did last summer. We're going to repeat it again this year. And from this data, we now understand, yes, the population looks healthy. It's well balanced across the size ranges. So we're fairly confident that we've got a healthy population, that the spatial pattern of use shows that it's concentrated largely outside the MCZ. So I'm beginning to be reassured about the status of our population. But of course, there remains huge uncertainty with this species and what's going on. So I feel there was a bit of a challenge to us and to everybody about using the data better. I don't like my first bullet point. I felt there was a bit too mealy-mouthed. It was just a bit too precautionary. But I think that the second bullet point, we need to know where these species are. A marine protected area is about directing resources, it's about directing effort. So we have to be a bit clever about where we direct our monitoring, and that makes sense. And also makes sense that we're focusing on the critical life stages of these species. Fisheries research, conservation research, they need to talk to each other better. They just don't at the moment, but it's not, I think the fact there's different laws and different policies just doesn't help. So we know that the fishery is healthy, but we need to make sure that the conservation status are speaking to that. But lastly, the other obvious point is that we need to connect knowledge, ecological knowledge of fishermen. You know, Mark, I guarantee, will know more about crawfish than anyone else here. What's going on with that population? How do we take that kind of knowledge and insert it into the way that we manage the species? Is that okay? I've done about 10 minutes. Thank you much.
Thank you very much, Tom. More than okay. Spot on. Right, next up, Libby West from Natural England. As said earlier, she's going to be talking about how we can incorporate telemetry data into national policy with something about fisheries management plans.
Actually, through no planning on my part, this talk fits in really nicely. So who are Natural England, first of all? Tom just covered that nice divide between sort of what we think of as classical fisheries management and nature conservation really nicely. And Natural England are the government advisor for the natural environment in England. And our purpose, you can see on the slide, but it's about thinking about present and future generations. We're interested in a couple of things. We're interested in the impacts of fishing and the impacts of lots of other marine activities increasingly, especially offshore wind and all of the issues that brings. Traditionally, Natural England are interested in the impacts of fishing on the environment. And does the natural environment include fish? Now, it probably shouldn't really be a question, should it? We know that fish are part of the environment. It's really important, but the way we manage them, as Tom just really nicely described, are quite separate to the marine environment. And there are some good reasons. We eat them. We want to keep eating them, and that will continue. But there are ways that we can bring these two processes together. So marine protected areas have been covered really well. So I just want to give a really high level overview. Our main job in a lot of ways is to make sure that MPAs are managed correctly, and we give statutory advice to the regulators as to whether we think management measures that are brought in for the protection are doing their job. This is the list of fish that are currently included in MPAs. It's a pretty short list. So what about the other 300 species of marine fish? Most data, as Tom said, is collected for sort of stock assessment purposes, and that's rubbish when you're trying to look at something at a kind of an MPA scale, or even actually at a local or regional scale. You know, certainly IFCA, formerly before Natural England, was at Devon 7 IFCA for a long time, and it's really hard to get anything meaningful from stock assessment data for any kind of regional management, which is fine because we don't manage stocks regionally at the moment, but maybe we would like to. So Natural England, along with a lot of organisations for a very long time, have been talking about something called an ecosystem approach. And there was a really nice description of it earlier. Ecosystem approach means a million different things. I think every academic, every practitioner has a different description of it. So I'll kind of talk a little bit about our interpretation of what an ecosystem approach might be. But very basically, it is bringing those two, the marine conservation world and the fisheries sort of science world, together a little bit more in some kind of practical way. And the key thing about an ecosystem approach, it's not all about science and it's not all about stock assessments. It's about how people are incorporated into that process. And I think that's really, really key when we think about projects like Fish Intel. So we've got our own definition in the UK now of ecosystem-based approach, thanks to our new Fisheries Act. And things can be called from ecosystem approach to ecosystem-based approach to ecosystem-based management. We seem to have just gone for the one with the longest possible acronym, which is ecosystem-based approach to fisheries management. So I won't keep saying that throughout the talk. I will just refer to it as ecosystem approach. But it is this idea of the fusion of parallel paradigms. And I go back to a really old UN FAO document that was written about 2003 that is still the most useful kind of paper on what this might actually mean in a practical measure. So yeah, fisheries management focused on stocks achieving MSY, generally considered single stocks, although we're starting to move away from that. The management's really centralized because we need these big data collection protocols and we need everything to be the same across those areas to be able to do the kind of statistics we need to do. But that means that it's quite often quite feels very far removed if you're collecting data locally. There's very little consideration of like the linkages to habitat or of fish's role in food webs in general. That is improving, definitely. There are definitely moves to improve that, but it's a general rule. Ecosystem management, we tend to focus on marine protected areas, on these little pockets that do really important things but can't fix everything. Tends to consider almost everything but fish. We just have to go, oh, fish are really hard, we want to fish them commercially, so we take that out and we deal with that totally separately. And again, don't really think about the fish's role in those ecosystems a lot of time, apart from those really specific examples of a few fish and a few MCZs and SACs. So, as I said, ecosystem approach means a whole load of different things. There are several hundred books written just trying to decide if you can come up with one definition, and the overarching thing is no, we probably can't. But as a general rule, to give you a flavour, it's about managing ecosystems within the limits of their functioning. So we want people to be able to use the ecosystems. We have to, completely accept that people are going to want to fish, they're going to want to build wind farms, but we need to make sure all of those activities are happening within that kind of threshold of ecosystem functioning. Preserving biodiversity, acknowledging species interdependence, so acknowledging fish as part of the environment. People are a huge part of it. It's about participation. I mean, Fish Intel is a brilliant model for this. The kind of diversity in this room in terms of roles and government and industry is a really, really good example of sort of ecosystem-based approach to science. It uses scientific knowledge, but also local knowledge. We need to get better at incorporating local knowledge into the way we manage things. But again, that's to do with spatial scale. People's local knowledge is local. That doesn't fit neatly into these big kind of stock assessments that are at the scale of the entire North Sea, Channel and Celtic Sea. And then it's about integration. It's about people like me who are kind of trying to interpret this and kind of make it see into policy, making sure that we're kind of doing our job to make those things, as Tom said, we've got lots of different laws and they're all separate. How do we integrate them a little bit better and ultimately decide? It's a matter of societal choice. What does society want? Do they want fish to eat? Do they want low carbon power? I think, do they want kind of biodiversity? And we know that we want all of those things. When we make trade-offs, they need to be based on societal choice. So it's a very broad concept. So how are we doing it in the UK? So we've got our Fisheries Act, and there's a lot of excitement around the ecosystem objective. You'll hear a lot of people say the ecosystem objective is the ecosystem approach, because that's what the Act says. I'd actually argue that it's very much all of those Fisheries Act objectives. You need to implement all of those to get anywhere close to an ecosystem approach. We've got the joint fisheries statement, which I recommend everyone goes away and reads, or at least if you want to just read one thing, about the first 6 pages of the Fisheries Act is really, really useful. And then we've got fisheries management plans. And I should say Natural England isn't leading the development of these. These are different co-management models being tested by DEFRA. But we are engaged in that process with different degrees to different fisheries management plans. But the overall kind of aim of the FMPs is to be a bit more stakeholder-led. A lot of them are being led by industry and start to kind of develop this ecosystem-based approach. Something I think we're doing well in the UK with our new legislation is acknowledging the importance of the environment on fish and fisheries. So we are saying fisheries are dependent on a healthy environment. Still don't think we're doing a very good job of acknowledging the role of fish in ecosystems more widely. So this is an image that John Davies from DEFRA gave me after his Coastal Futures talk. And what can we expect from fisheries management plans? Well, the first ones will be published. They're in a review process at the moment. BAS will be published this year. I think we should expect that probably there will be some inclusion of kind of wider environmental interests maybe in some of these first FMPs. I think we've got to see it as a long-term iterative process, which is good because it gives projects like Fish Intel time to input into these processes. There will be public consultations. Again, I'd urge Fish Intel to think about, the data that you've got and how that could go into those consultations. There are lots of evidence gaps being identified by FMPs, and we have DEFRA evidence in the room. I won't point because everyone wants to go and speak to them. But, it's going to take a lot of partnership working to fill those evidence gaps. It's not going to be for DEFRA alone to kind of work through that very long list. So there is an awful lot to contribute to, and it's a really, really good moment in time for projects like this to kind of input this kind of data. So the thing I'd say to anyone in this project is spell out what your policy lessons are. Make it really clear, because the people working policy at the moment are so busy. They don't have time to kind of really dig around for the meaning. So link it to specific Fisheries Act objectives. What are those lessons we should learn from telemetry? And I've had a little stab. So I think the more detailed information that you get from telemetry on spatial ecology becomes really relevant when we think of the people angle of fisheries management plans. If we're going to incorporate what local people want to see from management, then we need to have that data at a much finer resolution. The information on habitat use, the interactions and productivity is really, really interesting. One disappointment from a personal level is that the draft JFS mentioned essential fish habitat multiple times. The published JFS doesn't mention it once. It's fallen out of it. So that's a big job for projects like Fish Intel to keep pushing. So the improved understanding of food webs and trophic function, I think basically we're still losing that argument about fish being part of an environment. We can't keep managing them separately. And then ecosystem-based fisheries management to ecosystem-based management, this awful terminology that we've come up with. Again, it's thinking about those wider implications for telemetry, because fisheries generally still have the biggest impact overall on the marine environment, but that could be overtaken fairly quickly by offshore wind at the scale that we're looking at to meet the kind of government's energy strategies. So different kinds of pressures that telemetry could be really useful for informing in the future. You know, we don't really know what electromagnetic fields do for a lot of species. And we're going to have offshore wind farms all over the place. So if we've got existing arrays and we've got existing data, then looking at how fish change their behavior in relation to noise from construction and electromagnetic fields. And then real life information on how cumulative impacts, kind of how all of these different things mash up, kind of change fish behavior. And then the changing importance of locations and habitats over time. Thank you.
That's brilliant. Thank you, Libby. So it's back to us, is it, Libby? The ball's in our court. Thank you. Right, our next speaker is Lydie Couturier, and she will be speaking in French. Where is she? There she is. Hi, Lydie. And she actually, that's a nice segue from what Libby was saying, is from France Energy Marine, and so she'll be talking about some fish tracking in relation to offshore wind.
I'm sorry to speak English with an English-speaking audience, but hello, everyone. My name is Lydie Couturier. I'm a researcher at France Energy Marine. And as Emma's just told you, I'm going to tell you about monitoring within offshore wind farms and working on the Fish Intel and another associated project called Fisher that I'm going to present as well. So setting up offshore wind farms is obviously going to have an impact on the marine environment and habitats. We have some fairly well-known effects like noise and sound and electromagnetic fields, and the movement of substrates that in fact could create a reef effect and change the habitats. And the image you see below here shows in fact the consequences, potential consequences, with individuals who are going to make use of this new habitat in different ways, modification as well in the connectivity of areas used by fish. So the challenge here resides in our ability to detect these aspects, and it takes some basic knowledge. In fact, well, that means identifying the essential habitats of fish and how the individuals, in fact, create connections between these habitats. So we need methods that are appropriate and some innovative protocols as well. And they have to be relevant. They have to be developed in such a way to meet these needs. And we have to ensure that offshore wind farms, in fact, are part of larger networks of monitoring. And so this is the framework in which the Fish Intel project. The two projects are involved, Fish and Tell, first of all, with large-scale monitoring, including offshore wind farms, and then Fish WF, where the monitoring is more applied to species that move more or less and might be more sensitive to electromagnetic fields. So Fish OWF, Fish OWF, in fact, brings together public administrations and organizations, institutes like Ifremer, associations like the Pêches, small companies as well, and developers of offshore wind farms. And with all these partners, the objective is to develop an effective monitoring strategy that will enable us to identify and assess the effects of offshore wind farms on fish and large crustaceans. So Fish OWF, there are four main aspects that are studied and groups, work projects. There you see in the green square, if you can see it, the fine scale modeling or monitoring of movements of fish and see how they behave around structures like connection cables. And the second aspect involves how the individuals, in fact, occupy these areas and what their movements are within these zones. And we're using 3 offshore wind farms for that. The little green circles that you see, one at Calvados, one at Saint-Brieuc, and another at Saint-Nazaire. And we also have some floating structures that are serving as proxies for the future offshore floating wind farms. You see the orange structures. There are 2 in the Atlantic and, 1 in the Atlantic and 2 in the Mediterranean. And a fourth aspect, fourth work program, a package, excuse me. And this involves, in fact, the networks of acoustic telemetry monitoring and work both in the Atlantic and in the ResMed network in the Mediterranean. So we're looking at the sensitivity to electromagnetic fields as well. That's the case for certain fish and certain large crustaceans who are going to respond in a certain way. However, we're looking at species that tend to aggregate more and congregate, or those migratory species. So very briefly, the species that we focused on, this is really in the Atlantic and Channel areas. We put external tags, it's like piercing, and we have 4 species with internal tags as well. So external for these rays, internal for the sharks that you see here. And finally, an external tag on European lobsters. So all of these, in fact, were able to benefit from the annual surveys and campaigns run by Ifremer. Now let's look at the farms. Here you have maps of the various wind farms that we monitored. Oh, she's moving away. The colors represent the different types of habitats. So we have the same color codes used, but the one at the bottom, I was not able to obtain the files necessary to provide this information. So you have coarse sand in yellow in the various parks. And in fact, this was a decision made in consultation with the fisheries committees, and we wanted to position our receptors with respect to the activity in these wind farm areas. And so some of them have active fishing activities taking place. Others, you have a wind farm that is simply being constructed. And then we have one that is operating already. And that's much more interesting in terms of the science. If we zoom in on the wind farms, that of the Calvados in the Seine Bay, in fact, will be constructed as of late 2023. We were able, though, since the end of May 2022, deploy 9 receivers, and we were able to tag 43 individuals. There are the graphs. I'm glad that there were explanations provided earlier. Each line or dot is an individual, and we see their movements. And then I try and use a color code representing the different sectors in this area. In this zone, you can see there's a variety. Some individuals were released and detected, and then we never saw them again. And we see that there are a number of detections around the floating buoy that we put in place. And then you have some movements up to 30 nautical miles away from or distant from the wind farm. That's interesting. And then we had one tagged individual who stayed there. That was the star of our group. Now, very quickly for Saint-Brieuc, for example, this park, in fact, was under construction since January of 2022. We were able to deploy 16 receivers in the northern part of the wind farm, and we were able to mark 50 skate and shocks. And somewhat to the west of the park, 2 poor beagle shocks. So this shows you where these individuals were found, where they were detected. There weren't a lot that actually were sensed. We can see in the northern area of the wind farm, at least, they tended to navigate quite a bit, to move around. And there we can see that individuals are occupying these habitats. Maybe not as intensively as another example I'm going to show you. Those are the 2 poor beagle shocks. We could only tag 2 of them, but we were able to monitor them and follow them. They were marked here. The detections you see, I put arrows there to show you. This corresponds to the graph we saw earlier. And the red and khaki colored drops correspond to where they were marked. And then where we were able to detect these 2 individuals, we were able to detect them in the Saint-Brieuc wind farm. So they seem to have come back, and there was the female who stayed several days in the area. And then finally, what we discovered last week, 2 bluefin tuna marked by Lucy, in fact, and they were detected several times over a period of practically a month, where we would have several detections. In Saint-Nazaire, now this is on the Bay of Biscay, and the park is reaching completion of its construction phase. Well, in fact, it reached completion in December 2022. And we were able to tag 63 individuals. And just to show you our initial data here, each individual here is shown by a dot, and then the color codes apply to the receivers that you see on the left. And you see areas that are stay very much in the area around the orange receiver. And then you see others that move. Some stay in the area, and others move around quite a bit. and other individuals that we were able to tag, like the two porbeagle sharks. In fact, we were able to discover also a sea bass that had been tagged in the Côte d'Armor region of northern France. Northern Brittany was also detected in Saint-Nazaire. And the lobsters, in fact, we've detected, we look to see whether they've moved much or not in this area. So I just wanted to give you a glance at the data we've been able to collect. And the next step is going to be to link our detections with the environmental parameters. And what we are going to do is try and validate this method for fish populations within wind farms. So what we're hoping is that we'll be able to deploy this over the long term for monitoring, and this could provide help to the fisheries sector, and there'll be new methodologies and novel methodologies like acoustic telemetry. And if you are interested, if you want to learn more, information is available on our site, both in French and in English, so no problem there.
Thanks very much, Lydie. I should have said, actually, Lydie was the inspiration for Fish Intel. So we are very, very grateful that she came to us many years ago now, actually, with that first idea. Okay, the next talk is another double header, another French-English challenge. Collaboration, Katia Frangoudes and Alice Hall. So Katia is from the University of Brest. Alice is here from the University of Plymouth. Over to you guys.
So here you have the human dimension of this IMBFM. So our action in this project was more the relation between the different type of stakeholders. Here in UK, you are lucky. You have everybody in the same groups on the level of IFCA, from what I understood. In France, is more complicated because we have the fishermen, commercial fishermen and women, and the recreational fishers, which is completely different organizations and people. Then we have the administration, then we have the conservation, and then we have different, but we never meet. So at least at this level, you are more lucky than we are. So our role with Alice here in UK was to meet the stakeholders. We organize meetings. Our first objective was to gather fishery ecological knowledge to bring it on the IMBFM, in the future IMBFM, and also to define with them more or less where we are deploy the receivers. From their opinion, where it's the most important places that we deploy this receiver. And oh, sorry, I move a little. And then we had another action, which was the habitat mapping that Alice will speak about it, and the ecosystem services. These two actions, they help us to build more or less the web atlas that we will show to you what it is. And then we have this issue of IMBFM that we will discuss this afternoon also with some of you. So I just start to speak. I always have problem to follow the PowerPoint, but anyway. So we are we had this meeting, the first meetings where we ask fishers to speak us about their activity and where they think that they are the nursery areas, the essential fish habitat according to them. And then we done a small survey in the two countries concerning the health and well-being of recreational and commercial fishers. In France, we follow another way because we had a little problem to tell people to speak about their mental health. The word mental, it's a culture. We don't think that, so we spoke about more well-being and so on. And we ask also the current view about their views about the current and the future management, if they are satisfied or not, and so on. So for that, this part on the health and well-being, we produce a report for Interreg, of course, but this report is an open access document that we can find, we can help you to find where it is. It's on Ifremer in France website. And in the other hand, we done a survey with recreational fishers. And this survey, sorry, it's in French, the document, but this document too is online and it's an open access document. So now I leave Alice present to you the
web atlas. So the web atlas is basically a collection of maps that we've created and we've consolidated from lots of different areas. So we have habitat maps, so UNIS level 2 and 3. We're going to go into more detail in a minute on how we've done them. Ecosystem service maps. So we've created maps for food, healthy climate, primary production, regulation of water and sediment, formation of species habitat, formation of physical barriers, and carbon sequestration. So all of those layers are on the web atlas, along with the marine protected areas for both the UK, France, and Belgium, the renewable energy sites, the aggregate extraction areas, and also any water quality data we have, so from the WFD. So all of this gives you kind of a picture of how potentially healthy or impacted the ecosystem might be that you're looking at. So we're going to give a live, no we're not, I'm going to explain the maps first and then give you a demonstration. So for the habitat maps, you can see two examples here. So we've got the channel wide on the right and then a pilot site on the left. So the whole of the channel we have got a habitat map for. This is a combination of survey data and modelled data. So where there isn't any actual data, the habitat is modelled. And if we've got survey data, then you've got the highest level of kind of confidence in what that habitat is. So this is all on eModNet. So most of this data came from eModNet. We're also going to supplement it with some habitat surveys we've done in-house as well. So areas where there isn't data, such as kind of the mussel farm and some of the areas in the MPA in Lyme, we're just going to kind of update those habitat areas with the kind of most recent habitat. So on the maps here, you can see, so each colour is a different habitat type, so different UNIS level. We've got it for UNIS level 2 and 3. And this is what we use to kind of overlay our tracking data and also the ecosystem service data as well. So here are the ecosystem service maps. So I've just got one example to show you here. This is for food, so the ecosystem service of food. Again, you've got channel wide and you've got the pilot site, so this is South Devon. And you can see the darker areas is a significant. So this is the potential ecosystem service value from that habitat. And this is all done from the Rees et al. Paper, who's done it previously on the north coast of Devon. We've basically upscaled it for the whole of the channel, which was a mammoth task and had lots of help from Tom Woillez for that one. So you can see we've got the potential ecosystem services for the whole of the English-French channel. So I should say the pilot sites are all the hashed areas on the map. So that's the kind of unique pilot sites that we've been working within. Okay, so now I'm going to demonstrate the web atlas for you. So this is the web atlas. So it's completely hopefully user-friendly, hopefully easy to use. So when you open it up, it will look like this. So you've got the map here, and you've got all the different layers on the left-hand side. So if you click, you can scroll up and down. If you click the little circle next to a layer, keeping you in suspense. Ah. What could go wrong? I knew this was going to go wrong. So if you were to click a layer, it would appear on the screen. You can have multiple layers. You can layer them all up. And if to get the key, you press the little symbol. So if you want the legend for each layer, press the little cog, and the legend will come up on the left-hand side below all of the layers. Is it working? No. So this is hosted on the Ifremer Sextant web page. So when you go on it, it may be in French, but it will automatically translate it for you at the top. So it will say, would you like to read it in English? And then it will translate everything for you into English. So you can either have it in French or English, whatever language you prefer. We can show you afterwards if anyone would actually like a working demonstration. And new technology wouldn't be my friend. So we'll move on. Okay. So Libby did my job very well earlier in describing ecosystem-based fisheries management. So I'm not going to go over that again. But basically, it's looking at the entire ecosystem. So not just one species, looking at the knock-on effect it has, and also the different stages of a species life. So not just as an adult, but also as a juvenile, breeding grounds, spawning grounds, and just trying to encompass the whole life stage of that species and the ecosystem as a whole. So we have got a workshop this afternoon, which we're going to be discussing this and how we can potentially implement EBFMs and what the potential barriers might be to that at the moment. So I'll pass back to Katia.
Yes. So when our colleagues from the work package 1 and the work package 2, the person that they were spoke to you before, they finished their work. So we return back and to meet again stakeholders in both countries. And our purpose is to have their feedbacks from what they heard from the results of the project, but also mainly to co-construct with them these roadmaps for EBFM for each species and in both sides of the channel and so on and so on. So what we just put on this slide is just what came out during the meetings in both countries. So as Erwan and somebody else this morning spoke, tell you is that bluefin tuna and sea bass and crawfish and black bream, we see that the stock is coming back. It's recovered more or less. I don't know if it's MSY now or not, but it's not my business. People observe that, and we still have this problem with pollock. So for which we need to act. Erwan this morning spoke, and in France, but here too, a lot of fishers, recreational or commercial, they told us that we need to act, we need to do something. So then we asked them, but for you, why we are almost in this, we have problems with this species. So we try to discover, in fact, in general, for all the species. And many people spoke, okay, I know better what we heard in France because I was present, but Alice, she was present here, so she can confirm or give other wording of people. But it seems that there is a fisheries pressure due to innovation. In fact, now trawlers can fish almost everywhere. They can go the limit of the rocks, they can go very close and so on. The regulatory system, and I think Erwan this morning spoke about it, and yesterday we had another meeting where we spoke, is that when you ban fishing for one species, the fishing effort is going somewhere else, is transferred to another species. I mean, we don't tell something very new, but it's an observation, and we know it's the same again with territorial ban. spatial ban. We know that if we close one area, we move somewhere else. I think what people doesn't know, and they don't want to know, is that the sea, it's an open space, yes, but each fisher has its own territory and fishing area. And so it's very difficult to move from one place to another place because somebody else is somewhere else. So that's why we must be careful when we decide this. Anyway, the other story is that we heard in Normandy, in fact, and they spoke about wrecks and the destruction of, since the Second World, these wrecks start to be destroyed. So the idea is here, I don't know, we cannot put any, I think the society today cannot accept that we create artificial reefs like we done 20 years ago in some parts of Europe. But the question is wind farms something that can be a new artificial reef? We don't know. It's according to the countries if we can fish or we cannot fish. So, and we heard about pollution, quality of the water, estuaries, nurseries, and so on. So for then they told us something about the future management. So in France, but Lucy this morning also spoke about the tuna, the bluefin tuna. The people, fishers, they claim for allocation of quotas, and we know that we have limited number of quotas. In France, for sea bass, we have two different regulations between the South Atlantic and the Channel. So the Channel is more restrictive regulation, and our fishers ask to harmonize the two regulations, taking the Channel, which is more restrictive, as rule. So crawfish, it seems that more or less is going positive. Is positive, even if Tom said that we don't know too much, but we think at least in our cases. And then we need to do for Pollack, we need to produce knowledge. So acoustic telemetry can contribute to that. They spoke about restore habitats, especially the kelp, and this is something that you were thinking to do, I think, here in UK, and increase the minimal catch size and apply at least seasonal restriction as we have in France about season for sea bass, and regulate catches of recreational fishers. But recreational fishers in France agree too. So we hope that we will reach an agreement in the near future. Thank you.
Thank you very much, Katia and Alice. Okay, we've come to our second question session. So if the other speakers from this second part of the session would like to join us on stage. Again, I'll start with questions from the room and then go to the Slido. Jean-Luc. And then Randy. Have we got microphones? Alice, have you... All right. Yeah, make her work. So, Ada, it's just Jean-Luc at the back.
I've got a question for Tom, Lydie, and everyone else. There is a thing called the Habitats Directive, which we will bloody well lose in the UK because we're idiots. but it is a fantastic piece of legislation that combines fisheries and conservation policies because it talks about typical species in the features. Typical species of features are fish, not just how hard a rock is. We don't need the favorable conservation status of a rock or a grain of sand. We need the biology associated with it. So can we use that thinking that is in there and with all other policies that Lydie talked about to just move where we can with this science that is emerging, that there are some sites that are clearly important for biodiversity and for fisheries management?
Habitat regulations, favourable conservation status and typical species. So Natural England are doing work on both. They are actively looking at both of those things. They are things that have been being looked at for about 15 years now, as you know, but there is some progress. Personally, I think when we think about ecosystem approach, I still struggle with habitat regulations because it's still only a limited number of species. But yes, I think there is more we can do with the habitat regulations. So I think if we can start to define, I think the work that we're doing, for some reason, we're erring away from typical species and it called something slightly different, influential and structural. We're making it longer, of course we are. Yeah, so there is work underway and I think it could take us a step further, but I don't think habitat regulations will get us all the way to an ecosystem-based approach.
Mention my name, so I'll add my toughest worth. You know what? You know, we can do better. And I always felt with the even though that was the case, Ollie, wasn't it, with the habitats regulations, that the way it was used was never like that, or very rarely like that. So you know, I hope we can use those mistakes and things that we didn't do very well and do them much better and try and create this kind of holistic environment that you described. So that is a good point. Yeah.
Any other questions from the room? Oh, yeah, sorry, Randy. Yep.
They're all fascinating talks and lots to pick up after late this evening. A question for Pete on bream. Yeah, the female site fidelity stuff's kind of quite interesting. And I guess sort of hanging around for 10 days matches a lot of the direct observations Matt Doggett did for males in terms of kind of nesting time. So yeah, it was just a thought as to whether potentially for next year and whether you're thinking about that. The resolution of the VPS is 1 meter. Okay, so I guess with bathymetry, you can map the nest. So whether for the next nesting season, you could map the nests and then you could track resolution and movements between nests. Because I guess the interesting bit, we know that males guard a single nest and there's sort of potentially 2 spawning cycles. So the fish will disappear for 3 weeks before there's a second wave of nesting. And it'll be interesting to get your thoughts on whether you see that second wave with the female movements as well. But I guess it's to answer that question of whether sort of females pair with males or have a small range of nests they may contribute to. So yeah, just your thoughts on potentially bathymetric survey to map the nests and then match that with the telemetry to show how females move between those nests and have those relationships with the nesting males. Fascinating.
That's definitely on the agenda, Randy. And in fact, one of the reasons we chose this particular site is because it is already multi-beam data for that area. In fact, every year it's within a survey box that the tarmac CEMEX, if you don't know, their aggregate dredging within in the vicinity of the Kingmere, and one of the compliance aspects of that is that they run multi-beam surveys to map bream nest sites within the Kingmere. So not just next year, for this year that I've just shown data for, we do, when those surveys are processed, potentially have access to nest maps, basically, so we can overlay and sort of try and match up fish positioning to nest positioning. It might not, it's never that simple, but it's certainly it's something we can try, definitely.
Brilliant. No, it'd be really interesting to see, like I say, the relationship of the females to the males and number of nests. Great for the work.
Thanks, Randy.
Any other questions from the room? Slido.
So we had a couple of questions come in on crawfish in regards to juveniles, whether any juveniles were tagged, and also whether any differences in crawfish movements were detected between different sized individuals, also with relation to potential juveniles.
Did you catch that?
I have a first answer. In French. What?
I'll answer in French. What we know about juveniles, juvenile crawfish, because we had a similar study with conventional tags and not acoustic tags, is that they seem to cover long distances. There was a question asked about sea bass and whether or not it could cross the channel. We can answer for crawfish, juveniles, immature young crawfish do cross the channel. And we had one that had been tagged by French fishermen on the French coast, and it was found on the other side. And it would seem that juveniles move around more than the adults, to be confirmed. If you'd like to supplement that.
So in contradiction to Marshall, I mean, the crawfish that we tagged are obviously a certain size to cope with the weight of the transmitter, and across the size range that we did tag, we couldn't see any difference in their movement characteristics, and the main difference was, as I mentioned, between whether they were male or female. But yeah, but I mean, we've only got a snapshot in time so far, and as I say, we collect more data, we might get more data which complement what Marshall said.
So on for wind farms, whether floating wind farms could represent a fish aggregation device. Did you hear?
Yeah, it's where a wind farm can represent
Can
aggregating
represent
device.
a fish aggregation device, so where fish collect. Yeah.
Yeah, it can.
Yes, the answer is yes,
No,
they
but
can.
That's the question is how the fish are going to be using this this new habitat. Do they stay? Do they just come and hang around and leave? But then they come back, or do they just come by because there's something floating and they're curious and they just swim by? A whole lot of questions that we don't have. But yes, that's what we expect anyway. And that's what's observed around the buoy we're monitoring as well. It's a fish are aggregating.
Are there any more burning questions on Slido?
Unless I'm not sure that we've got time. There's quite a few questions on Slido, so I just wanted to say that we're not going to have time to get through them all. We did let people know that at the beginning, but yeah, we've had quite a lot of interest, but just unfortunately not time to get through it all. We can get back to people. Yeah, we can share the questions with the speakers, and they can get back to people.
Okay, lovely.
Thank you.
All right, thank you. Okay, so is Fred here? Okay, wonderful. So our next speaker is we're now crossing time zones. He's joining us from Dalhousie University in Nova Scotia. Fred Karleskind is the executive director of the Ocean Tracking Network, and it's really great to hear. Oh, there he is. It's really great to hear from him today. We saw him recently at an Impact conference in Canada, and it was really amazing to hear what they're doing over in Canada. So it's brilliant that you're here with us today, Fred. Can you hear me? Read your lips, but I'm not wearing my thing, so I'm going to go plug in, and I'm going to hand over to you, Fred.
The one thing we did not get to test was the microphone in our practice, so I do not know if this is working.
Yeah, we can hear you, Fred.
Okay. Now, let's see if we can share the screen. And very good. Well, I thank you all for the kind invitation to be with you today. I wish I was there with you. This is very heartfelt. This morning at 3 a.m. when I got up to come in and log on to the conference, I discovered we'd had a snowfall and I had to shovel the driveway to try to get out. So that was a lot less fun, but I did get on. I've been listening with great fascination to everything that you have been saying. It has been a spectacular success, and I congratulate you all on what you've accomplished up until now. I was asked by Emma and Amy to come in and act a bit as an eminence grise, offer some pearls of wisdom. My wife hasn't stopped laughing at that particular concept at this moment. I also know that most of the wisdom and things that I have learned and think that I have discovered has been discovered by others who are far more eloquent than I am previously. So in the course of what I will be doing with you right now is showing you a few quotations from some of these brilliant people that will help structure the discussion that I'd like to have. And I believe that in order to understand where we're going to be going in the future, we also understand a bit about where we've come from in the past and dredge up some memories of you for the early parts of the Fish and Tell program. So the first quote I wanted to share with you is coming from a Nature editorial fairly recently in 2021. It reads, "The future lies in standing on the shoulders of crowds. Forget standing on the shoulders of giants. Today, it's all about collaboration." There are many reasons for this. Some of it stems from the fact that the problems we now face are very big and they surpass the capability of one individual or one lab to address. Others are that we're highly technology dependent, and these technologies are expensive, and no one group can afford to put all the technology out, especially in the global oceans, that would be required to answer the questions. So we are moving forward into these kinds of structures that the Fish and Tell program created, this network of talented investigators that are addressing common problems. Now, my next two quotes come from Samuel Langhorne Clemens. Most of the world knows him as the American humorist Mark Twain. The initial insight here is that the secret of getting ahead is getting started. Thinking back over the Fish and Tell program, thinking back over what it has taken to bring together all of these investigators, thinking back on all you had to do in order to park other things, in order to put the time and the effort into building the Fish and Tell program, that getting started component is a really critical step that you took and succeeded brilliantly with. My second quote here comes from the American boxer Muhammad Ali, who most people would not perhaps think is a brilliant thinker. But this quote is about how it's not the mountains ahead to climb that wear you out. Rather, it's the pebble in your shoe. Here, my point is that you were operating within the context of Europe and European funding agencies. Within those particular agencies, there are some things that appear very small, but are very important to the European agencies that if you don't actually accomplish and deal with, things like reporting, things like financial management, things like answering queries that come forward from your agents. If that is not all done, the whole apparatus comes to a screaming halt and none of the work that the great scientists that are working on get done. So here it's a shout out to your host institutions and the Fish and Tell for meeting all of the needs that had to be met in order to do the reporting that kept the scientists focused in the areas where they're so very, very good at doing what they're doing. Now, looking forward, what are we going to need to know over the next years, and what can we do as telemetrists to help fill that in? Probably the very first and most major challenge we face is how to respond to a rapidly changing ocean. I cannot believe the changes that I am seeing, and here off of Halifax, Nova Scotia, just to give you one example, last summer in this temperate to Arctic environment under normal circumstances, children were capturing mahi-mahi off of the piers from which they used to fish for mackerel instead. So these are the kinds of changes that are coming forward. And with it, it's going to cause massive derangement and changes in species distribution. So we have a major role as telemetrists to try to help understand this. MPAs are obviously going to be a big thrust for the next 10 years. The when, where, and do they actually work and accomplish what they intend? Here in Canada, we made a commitment to protect 30% of our marine environment by 2030. This has had an actually nefarious impact in that there is a desperate scramble now to find areas, any areas, cobble them together to meet the bureaucratic target that has been set, the one that has been promised to the political community. It is not a coordinated design approach, which is one that is building reserves that are based on previous knowledge, well studied, and also on trying to link things to meet the needs of the animals that you're targeting at to do it. It's rather going to be a hodgepodge of little areas that are brought together, and that creates a nightmare in terms of monitoring them to start with, diffuses the very scarce resources that are available. And at the end of the day means that a major task we will be facing as scientists is trying to repair the damage that is done by fixing these areas in place when they're not doing the job that they were supposed to be doing. Endangered species will be another major theme. Things are not getting better. The COP meetings in Montreal very recently passed have highlighted again the rate at which we're losing species. We need to understand and bring those back. Biodiversity beyond national jurisdictions also on the high seas is going to be a major issue. Understanding animals and the connectivity between coastal areas and those offshore areas, and also the animals that are unique to those offshore areas. We have huge knowledge gaps that need to be filled. Overall, the implementation of ocean and coastal governments is really important. Many of your speakers today have already touched upon that in great detail, and I won't repeat that one. And you have also clearly identified the needs of stakeholder groups. Almost all of the projects that you have presented, that, or all of the projects, no, I take that back. It's all of them, not almost all of them, that you have presented are meeting the needs of particular stakeholders. And that will be an ongoing evolution, especially in the face of these other changes that are occurring. So plenty of work for us to do as we move forward into the future. In terms of what are the tools that we need to meet these future needs? Well, it starts with the ideas. We have to have those focal ideas, the ones that rally people to it and provide that intellectual stimulation as well as the practical and fundamental knowledge that will help us to fix the problems that we are facing. And we will need to get these networks of multidisciplinary researchers together and keep them together. We need the international funding mechanisms to fund international research. And this is going to be a huge challenge, trying to align the interest of different groups. Not to put too big a point on it, but the chaos that has been caused as we try to work through the Brexit decisions and trying to build together the networks that we've had in Europe before that were tightly integrated with Europe is a good case in point. It was also mentioned in some of the presentations earlier today that we have to be focusing on appropriate time and spatial scales. This is especially true in the face of this rapidly changing ocean. Yes, we're going to need short-term things, but we also need to have constant and ongoing monitoring so we can pick up larger and broader signals about what is coming at us. And the training of future generations of scientists, I think, is going to be a really critical issue. We have a decade of ocean science that has been launched right now, and there is a false sense of security developing, in my opinion, especially in the public, that at the end of this decade, all of the ocean problems that we face are going to have been solved. So I think we need to start managing those expectations to start with, but more importantly, get trained this future generation of scientists that are going to pick up the mantle from others like myself that are going to be forced out to pasture very soon and in retirement, and get them to look after those particular issues. Home institutional supports are going to be important, inculcating a culture within your universities, within your government agencies, that the kind of work that telemetry is doing is important and needs to be supported, displacing some of the traditional, for example, stock assessment approaches to doing things. And then we have this need to have new technology and data systems occurring. I think I have another slide about that one. So in terms of couple of slides, technology and evolutions, I just wanted to flag a new project that has just been launched. This is called the Straits project. It is there to serve, hopefully, the broader needs of all of Europe. What is occurring now is the placement of a series of acoustic infrastructures located within the Danish Strait. So anything going in and out of the Baltic Sea to the Atlantic and back again will be picked up. Sea monitor array that is across the Celtic Sea. We have a new line that is going into the Strait of Gibraltar and others that will be going into the Bosphorus and Dardanelles, looking at movements in the Black Sea into the Mediterranean and from the Mediterranean on out again. So those are going to be very powerful tools that will help pick up some of the broad scale, not the local scale, but the broad scale movement patterns that I think would be of great interest and use for a variety of investigators. Then we have other technologies that we have to develop. On the top right of the slide, as I think everybody is looking at it, you see a map. The map is showing in green and red a current deployment of acoustic receiver units. And what that is highlighting, and these are the acoustic receiver units that have been united through data systems that are cooperating, the Ocean Tracking Network, the ETN system. There are other groups in various other places with continued expansion into the Pacific Islands region right now and other areas like that. So those data systems are more or less seamlessly integrated and are going to be a powerful tool to let you understand in the face of this changing ocean how your animals are changing their distributions and as they're crossing borders and going places. Supporting them, maintaining them, that is something that we have to build into our core thinking. It is very rare that an investigator, when they begin to put a grant proposal in, thinks that 30 to 50% of my resources may need to go into data management, and I am going to budget that as part of that one. But that's some of the thinking that we probably have to evolve into. It was mentioned in one of the earlier presentations as well, that and by Emma, that the open protocol is a newly evolving component. So we've had different manufacturers of tags that may have been on the same frequency, but the way they coded their signals was different, and it meant receivers from one company could not pick up the coded signals from others. The open protocol is a shared program that all manufacturers have agreed to work. And the OPI is the open protocol itself. OPS is the open protocol for sensor tags that will go on the units. And now we have all of the major telemetry manufacturing companies agreeing that they will manufacture tags and receivers to the open protocol standards. This does not mean that these companies are going to stop manufacturing tags that operate on their traditional standards. And that is probably a good thing, because the capabilities of some of these tags that will let scientists answer questions that they want to answer are not supported by some of the open protocol systems. So it means that you can go back and forth. But the key now is to make sure that all of the receiver units that we have out there could at the least detect any tag that has open protocols. I am upgrading all of the equipment that we deploy in Europe as part of the Ocean Tracking Network as I speak right now, and I hope by spring of this year that all of that equipment will be fitted with the open protocol capabilities. So the Strait of Gibraltar deployments, the Baltic, all the rest of those will have that. And finally, I think we will be seeing an increasing use of robotics and autonomous vehicles in the near future. I am hoping to launch a developmental program soon that will be looking at trying to develop acoustic receivers that are rechargeable. And those rechargeable units can be recharged by marine autonomous vehicles, power sources basically, that would navigate independently and autonomously out to them, recharge the receivers, retrieve the data from it, and bring it back again. And that would usher in an ability to maintain telemetry networks at a much lower cost, much lower carbon footprint than it is right now, and at a much greater frequency, which I think would be the interest of almost all the investigators that are out there. So those are just some of the directions where I think the technology for the future is going. And in terms of Fish Intel 2, you guys have checked all of the boxes and everything that I have put up there. Congratulations on putting together your teams, on meeting the needs of your funders. You've got all the pebbles out of your shoes, and you've overcome the inertia. So you're out there, you've got a world-class team doing world-class science. I think you've identified the fact that your network has also got opportunities to network with the other networks that are out there. And that's probably going to be the next major development that we need to get done. Many of these small and independent groups need to be better coordinated, better integrated, and better sharing of almost everything that you're doing. And at the end of the day, I think it's also very clear that the knowledge that you're generating is very important for what we're going to be facing for challenges for the future of the ocean. So we have to keep you out there and doing this. So in closing, for me, it's congratulations to you all on what you've accomplished. Good luck with this future. And then it was noted that telemetry was going to generate knowledge by some of the speakers, and that is not actually true. Telemetry is going to be generating the data. It is you, the telemetrists, that are going to be pulling out the knowledge that's there. So you remain absolutely essential to the whole process. So with that, thank you very much and good luck with the future.
Thank you very much, Fred. That was really inspirational and giving us lots to think about and get excited about for the future. Network the networks. We're going to need to talk, aren't we? So thanks so much as well for getting up at 3 o'clock in the morning. I didn't realize that we had you from the start of the conference. That's really, really great to hear. I believe, Fred, that you're happy to take a couple of questions from our audience. Wonderful.
Absolutely.
So any questions for Fred? Tom. This is Tom Stamp, who you met at Impact recently.
Hi, Fred.
Hello, Tom.
Hello. I was just wondering, with the new Straits project, do you have any funding difficulties with the Sea Monitor project linking with the UK after Brexit?
There are always problems. However, the biggest one is the disconnect between the EU and the UK now following Brexit. And the tactical decision here and the great benefit that we have is that the LOCS agency is a cross-border agency, and it is eligible to apply into and represent proposals both in Europe as well as in the UK. So it is the bridge and it is the funding tactical mechanism that is going to allow the Sea Monitor line to continue to be funded and continue to be operated. So it was a fortuitous development, but one that we very much appreciate.
Okay, thanks. Unfortunate for us.
Any other questions for Fred in the room? I don't think Slido's caught up with this yet. This is a question from Marshall from Ifremer.
My question, it is a link that we develop with stakeholder. You know that it's really important when you talk about that. It is how you share your result in order to people are able to have the same level of knowledge in order to be able to exchange together. Is it that you have in mind?
Absolutely critical. It was also pointed out that you need to incorporate these stakeholders in your management regimes. Fisheries management has a little bit to do with managing fish and a very great deal to do with managing people. And if we do not have our fishing community agreeing to the management measures we put in place, they will just ignore them. For us here in North America, lobster is the big one, and the lobster fishermen are the best armed group of fishermen that you see anywhere else in the world. And they do shoot at you and they shoot at each other. So that's the kind of management regime you don't want to be getting into. So by bringing them into the process, by letting them know what the information is that you have and having them buy into that one, that's very good. And one of the things that we try to do in the Ocean Tracking Network is for as many of our activities as possible, charter commercial fishing vessels and commercial fishermen to assist. because what they become is not necessarily agreeing with everything we do, but they are ambassadors at a level to the fishing community. They speak in their association meetings. They can answer questions about what we're doing and why, and it dispels a lot of the false impressions, but offers up realistic expectations of what we can deliver for them. And that has smoothed over an awful lot of problems for us.
Great. That's like, thank you very much, Fred. I think I'm getting hungry for sure. So I'm going to let you go, but thanks again for joining us today. We really appreciate it.
Again, thank you for the invitation and congratulations.
Okay, so we're almost at lunchtime. First of all, would you all like to join me in saying thank you to all of our speakers this morning? I think it'll be absolutely brilliant. And then on behalf of all the partners from Fish Intel, I really want to express huge gratitude for you all coming today. And the day's not over, but I just know that some people are going to be dropping off this afternoon. So I just want to get my thank yous in today to make sure that I get them done. So on behalf of all the Fish Intel partners, we really want to thank the University of Plymouth for hosting, especially our funders. Interreg's actually in the room. He's been quiet at the back. But and all the other funders that have contributed along the way to all the work we've been doing, Natural England and IFCA and lots on the French side. We want to especially thank the University of Plymouth events team who have done an amazing job. I was expecting this all to explode, but this has been brilliant. It's we're not done yet. It's pretty smooth. I'd like to thank our interpreters who did a super job. And I just really want to now give a special thank you to the people who really organised this conference. So there's 4 people who have spent weeks, months planning today and made my life a lot easier. So Amy, Marta, Katia and Alice, please would you come up to the stage? The team have got a little present for you.